Oh, let's see. We have our speaker for today, Ibu Tri. Ibu Tri, his Dr. Tri Rinin is already here.
Hello.
Fortunately.
Yeah, thank you.
Good morning, Dr. Tri.
Yeah. Hello.
How's your
nice
day?
Yeah, it's great. Thank you. I hope you also have a good day and we are all still stay healthy because of this pandemic. Yeah.
Yeah. Yeah, I actually read some of your publication here. So you work with a lot with metabolomics, right?
Yeah.
So it is actually quite a novel concept for me. So I'm not really sure what I'm reading, but I see that it involves a lot of statistics.
Well,
as
a
I
bit.
see it.
Yeah, it's a bit of statistic
I-
there, but it's not so much. But actually, yeah.
Yeah, but I'm sure you will talk about it today in a lot more depth. So I hope you can introduce us to this concept.
Okay.
So, we also have our the secretary of our department here, Buher. Selamat pagi. We also have a lot of participants here. We have Wendell, Warda, and Safira. Tokir, welcome back. Tokir, how's your weekend? Right. We also have Nathaniel Woon. This one is from last year, I believe. We have also have one of our student, at least one of our student. I can't remember all of you, of course. Unfortunately, we have Farah over here. Also, Afrabias. Okay. Yeah, just here. Oh, yeah, right. So for today, we will be talking about metabolomics on natural products. It is actually a very novel concept for me. So I don't have a lot of things to say, and I'm very much learning like you guys today. Okay, can we start right now, Fitri?
I think it's still 6 minutes left.
All right. Okay. First, let me. Oh, Farah just received the essential oil package. Can you show us, Farah, the essential oil package that you get? Okay. Okay, wait. Where is she?
Good morning. I just received my
Morning,
essential
Farah.
oil from Asep. Here it is.
All right. Bumita, can you please stop share first?
Okay. This one.
All right, there it is. Wow.
I get for
What kind
essential
of
oil.
essential oil did you get?
I got rose hip, and then geranium.
Rose hip, right?
Peppermint.
Geranium.
And then cinnamon bark.
Wow. That looks sweet. Have you tried any of them?
What?
Have you tried any of them?
I haven't tried because I think I need a diffuser for yeah, for the
Right.
essential oils. Yeah.
Well, at least you can try to use it as a perfume, right?
Oh yeah.
So just make your perhaps your clothes and maybe your body to be smell a bit better, right?
Yeah.
Anyone else get the package other than Farah? Hello, Dia, perhaps? Okay. Fang Tan, did you also get the package? I don't know which. How many of you? How many of you did get the package? Have no idea. I forgot to ask last time. I believe it should be there should be 3 of you that get the package. Okay. Right. I think we get okay. Wait, where is did my camera just turn off? No. Oh, it's still on. I took it turn off. Right. Good morning, everybody. Good morning, everybody. So I thought my camera was turned off because of a bit of connection problem. So among us here, we already have our speaker for today morning session, Dr. Tri Rini Nurini Tias. Hello, Dr. Tri.
Hello.
Thank you for joining us today.
Yeah.
Thank you for
Welcome.
For sharing your knowledge about these topics today.
Yeah.
So, can you just give me a bit of this before going into much more depth about what actually is metabolomics?
Well, it's metabolomics is quite new actually, and maybe in Indonesia also not a lot of people is working with this. But basically, it's combination of chemistry and biology and also bioinformatics. Yeah. And the application is really wide. Yeah, you can use it for health, even for me. I work more in agriculture actually. Yeah, and then also you can also use it for industry. Yeah, and yeah, so it's a lot of application, even in the health, especially now they are using this to develop the specific for the drug discovery, for the cancer research. Yeah. So, and how can I say that? Yeah, we need a bit of knowledge in chemistry for sure, and then of course bioinformatics, and then also we cannot also avoid the biology. So yeah, you need those 3 things, and if you do not have those at least you have to master one of these, and then you can ask your college, your friends to help you, so then it works. Yeah, it's quite easy actually. Yeah.
So it's basically multidisciplinary,
Yes,
right?
exactly.
Right. Okay. Okay. Thank you once again from our for joining us as a speaker for today from our. I think we can start now. It's already 9 in the morning. So, all right. Once again, welcome to our virtual summer school program. This is our, let's see, it's our 8th lecture, I believe.
Mhm.
Yeah,
Okay.
our 8th lecture. This one is on metabolomics. So before we begin, let me remind you that all of this will be recorded and will be available on our YouTube channel. So if you miss anything, you can always check our YouTube channel and rewatch it, right? So first, let's before we start, I would like to play a video first on this on this topic. I hope you agree with you have no problem with this. Please the video. Okay. Please enjoy. Oh, sorry. I forgot to unmute myself. So I think that is the end of the video. It's just a bit of a glimpse into what we think, what metabolomics should be look like. Of course, Dr. Phil will further elaborate what kind of study is metabolomic actually. So, uh, so without further ado, let me first introduce our speaker for today. So our speaker for today is Dr. Tririni Noringtyas. She is a lecturer in Gajah Mada University. So she get her bachelor degree in Gajah Mada University, and then a master degree in University of Philippines. I think we have someone from the Philippines here. I forgot which one.
It's actually Los Baños
we'll
because
later.
a lot of UP there. So
right, and also doctoral degree from Leiden University in the Netherlands, with her research field specifically in metabolomics and natural product as biocontrol. We can see here it works with antimicrobe, biopesticide, and bioherbicides, and of course the discovery. Next. So this is some of her publication. We only take some of the most recent one. In here we see profiling analysis on pigmented rice. There's also a profile of agarwood, Gyrinops, also colchicine, I believe that I said correctly. Colchicine effect on ploidy level and morphological character of catocon pepper, and comparison of metabolic expression in root and leaf of resistant and susceptible tomatoes to root not nematodes. Okay, without further ado, Dr. Tri. All right, Dr. Tri, time is yours.
Okay, thank you very much for the introduction, for the kind introduction. Is my clearly received?
Yes, I can hear you.
Okay, so let me first share the PPT today. Wait a minute. Okay, this one I think. Sorry, I didn't put it in the first. Okay, here and so this one. Okay, so let me start. Thank you very much. Good morning for everyone who joined this virtual summer school today. Just like what already introduced, my name is Tririni Noringtyas, but most people call me Nuning for short. Yeah. And my affiliation is from Universitas Gadjah Mada. I started from Faculty of Biology, and then after my PhD, then I also joined the Research Center for Biotechnology and also doctoral study program for biotechnology. And now I serve as the head of the doctoral study program for biotechnology. So then if you are interested there, just contact me. Yeah. Okay, so today, first, thanks to the organizing committee who invite me to take part and to share my experience and also my knowledge in this summer course. Yeah. Today I will share one of my major works it's about metabolomics, and I changed a bit the title. Previously it was metabolomic study of natural product for biocontrol, but I think it's more for the development, because using metabolomics, actually you cannot directly get that biocontrol, but then it's helped you to at least the first step or the early step to go to the biocontrol. Okay, so that's why then I changed to metabolomic study for development of biocontrol. Okay, and let's go. Yes, so metabolomics, like already mentioned several times also in the video. I show here the picture is a bit blurred, because I just want to show that maybe it's quite new for someone or for some of you, and then some maybe already work a lot with this, but some maybe just heard it or maybe just read about the article about it, but not really clear. So that's why it's still not really clear. Okay, so then I hope later we can study more about this. And in terms of the interest in studying the metabolomics, if we look at these charts, when I put the query in the Scopus search there, then I put the metabolomic, I put the metabolic profiling, I put the, what is that? Metabolic fingerprinting as well, So then I got it at quite a lot of, it's not quite, but it's a lot of study or publication on this. But actually, it's first, it's not that long. The first coin of metabolomics is started in 1997, and then it's growing very rapidly up to now. If we see here that actually the chart, it's still going up. So it means that it still become the hot topic in the study. Then if you look at how is the interaction or where is the metabolomics place among the other omics, because we know that now the omic approach has become one of the important also approach in the life science, We know about genomic, Let me use this laser point, We know about genomics and then epigenomics, transcriptomics, and proteomics. Then we will have the metabolomics. So this genomics to proteomics is actually try to elucidate the central dogma, remember, from gene to the protein. So the whole study of those in an organism called is the genomics up to the proteomics, But then we come up with metabolomics, which is actually it's the ultimate of this process, because some of the protein may work or function as enzyme that may then will give you a bouquet of specific metabolites in an organism, and then we will study it as metabolomics. Those all, of course, it will reflects the phenotype, and then we call it also the study is phenomics. And then from the phenomic, we can actually try to elucidate all of the phenomic, how the phenomic can become like that. It's using those interaction with all of the omics. But today we will talk about metabolomics. So what is actually the metabolomics? So some mention it as a powerful platform for the global identification and quantification of low molecular weight metabolites in a biological sample. So I put some of those words in red because it's the important one. So first is global identification. So the goal or the aim of the metabolomics is try to identify as much as you can the metabolite, the low molecular weight metabolite, in a biological sample. So that's the main point. Or some also mention metabolomics as a comprehensive quantitative analysis of all metabolite in a given cell, tissue, or organism under a given set of condition. So here, the cell, tissue, organism is mentioning about the level. So in which level you want to study. It's possible that you will study in the level of organism. For instance, you want to compare between one human to the other human, or one plant to the other plant. But also, actually, you can also compare between one tissue with the other tissue. For instance, one of my work in the PhD is I compare between the mesophyll with the epidermis, yeah, and then also between the cell. It's also possible. Then the important part here is that the given set of condition. So here, because we remember that metabolomics is the ultimate part of the omics, which will interact with the environment directly. So that's why it will change a lot with different environment. So that's why you have to make sure when you do the metabolomics that you put all of the condition of your sample at the same treatment. Yeah, so that's why it should be really mentioned that it is in a given set of condition. So just like here we can see, let me, I don't know how to make it, okay, just leave it. I cannot see my, the up here, okay. If you see here, like there was a lady here that's eating noodles, yeah. And then we want to see how's the response to the omics. If this is the X is the time, maybe you cannot really see here, time here. And then the Y-axis is the response. Of course, you will not expect when you eat noodles, then you will have a mutation, right? So that's why there's nothing changed in the genomics. But then how about proteomics? Of course, some of the digestion enzyme will work there. So that's why it will have a change there. But how about the metabolomics? Then because you put a lot of things, a lot of compounds in your body, then of course, there will be a lot of response there. And by the time, it will really depend on when you take the sample, it will give you the picture of the metabolite. For instance, if you check it in here, so you will have quite a lot of different. But when, for instance, you check it in this part, you will see no different, right? So that's why time is also very matter in metabolomics. Take into account of this, yeah. The similar things is also happen in, for instance, in the plant. If you put a tiny insect in the leaf, yeah, surface of the leaf, and then of course the leaf actually will react with this. They will feel it, there's something there, and they may start to emit some of the volatile compound, try to talk with each other that there's some dangers there. So they produce a lot of metabolite. And those, it's part of the response of the this treatment. And that's why, and those of the all of metabolite that's changed, that's what we usually study in metabolomics. So that's the idea, yeah. So what is actually metabolome? So just give you the idea that, remember, the first you have genomic, so you study in the gene level, and you have a genome. When you study proteomics, you have protein and you have proteome, right? So here it's the same. When you do metabolomics, you have metabolite, then the whole set, the complete collection of small molecules in metabolite, that's called as metabolome. It doesn't matter whether it's indigenous or you put it something there, so and then it's incorporated in your organism or in your sample, or the exogenous molecules, it's still mentioned as metabolomics. Yeah. So it's still the part of the metabolome that you will study with. And of course here, the complete collection is really depend on which level you will study. You may also work in the level of cells. Part of one of my PhD also there is comparing between one cell with the other cell, type of cell, yeah. And then tissue, yeah, for instance, you will check maybe the epidermis here, compare with the mesophyll here. because they look different. So of course they will also produce different molecule inside. Organ, yeah, maybe plant roots, in the roots and the leaves, yeah, and maybe for human you will have heart, you will have liver and everything. And also you can also compare between one with the other, the blood of one human with the other, and etc. How about the size? I put it here, size with a question mark. That's the problem in metabolomics, because not like genomics that you can mention like, okay, the size of genome of Escherichia coli is maybe 1 million base pair or whatever, something like that, yeah. But you cannot say like that for metabolomics. Why? Because metabolome is really depend on your analytical tools. That's why some metabolists say it's ill to define, difficult to define with this. Yeah. Okay. And I will give you a bit of glance about metabolomics, because I believe that there is no one mentioning about this. So I will give you a bit introduction of metabolomics here. We will actually in metabolomics, there's two approach, the untargeted, then the targeted. Normally we use the untargeted, and then after that we will go to the targeted. Yeah. So what is the difference? Untargeted is the method measure as many metabolites as possible from a range of biological sample without any intended metabolism. So whatever is that, I will try to get the signal. I will try to identify as much as I can. That's the untargeted. I don't know any idea what I will have in my sample, okay? And then it's including sometimes the chemical and non-chemical, because sometimes I cannot identify it, but it's important. So just put it as X maybe. It's okay for the untargeted metabolomics. And this is the most appropriate approach to detect unexpected change in the metabolite concentration. So for instance, you want to compare the different physiological character of your sample. For instance, in my case, usually I compare it between the resistant or tolerant species or clone compared with the susceptible one. So there, I don't know what will come up. So whatever is that the metabolite, it will okay for me, okay? And then, but the problem with the untargeted metabolomic is especially the chemical identification, because it's not really easy, yeah, sometimes, and you will need a lot of time for this. How about the targeted approach? It's more to define group. You will do this especially for the verification and validation. Normally after the untargeted, I will go to the targeted approach. I will looking for if I have the chemical standard there. So I will do the absolute quantification to the series or a group of the metabolites there. So it's more for the validation. So if you do the untargeted, you will get a global story, a global picture of your sample. For instance, if you want to differentiate between this one, between this, the left one to the right one, so you will see here that, okay, there are some parts different, see? But you cannot really look at how is the detail of those. But you know there is some difference there. But for the targeted, normally you will just go to a certain part. For instance, from the untargeted, I see, okay, this is some different here. So then I can go to the detail, whether how is that the different, yeah, that will be the targeted one. So it's more for the validation. Okay, then, so which one will you choose? Yeah, I choose normally the untargeted if I don't have enough prior knowledge on the pathway associated to the certain physiological state. But then I should have a good background also in the chemometric or bioinformatic analysis. And you have a good support for the chemical identification. And of course, the most important also, you have enough time to do that because it's time consuming. Then how about the targeted? The targeted is if you already understand about the pathway, you already have the idea what is the group of the molecules or the pathway that may work, so you can go to the targeted. And you will there you will do the absolute concentration normally. And this just to give you idea that the metabolomics application is actually can be applied in a lot of field of study, like health. You can do the biomarker, clinical biomarker. Environmental, you can go like for the toxicology, the risk assessment. Industry, it's not a lot of people working for the quality control there. And for especially here, me, I'm working with agriculture. I work for biomarker, and also that sometimes it will be developed into biocontrol. So how usually we do with this, so we normally apply first the stress to the sample that we have, and then, for instance, I have a lot of collection of clone or maybe cultivar or varieties of certain crop, yeah. And then I don't know which one is tolerant, which one is susceptible of this for my treatment. For instance, like here the pathogen probably like blast or bacterial wilt, yeah. So then first I have to do the bioassay first. I have to find first which clone or which varieties is tolerant, which one is susceptible. After that, normally what I do is I compare the metabolome between the tolerant and the susceptible. From this comparison, then I will get the marker for the selection normally. So if I can pinpoint or I can identify which molecules or metabolite which is present higher in the tolerant, so maybe we can try to develop it as a marker to choose to select cultivar later for the breeding. Yeah. Or sometimes I can use this directly for the biopesticide, because if the compound is available commercially or it's abundant, I can isolate easily, for instance. So then we can try to apply it in the crops, and let's see how it will improve the character of the crops. And sometimes it also can be used for the direction for genetic modification. So here is like I mentioned, like what I already mentioned in the previous slide. Here is the non-targeted metabolomic first. Then from those, I will get the functional metabolite of interest that I got from the comparison, remember? And then for the biocontrol, normally we have to isolate or maybe synthesize, or if it's available already online or commercially, just try to use it for the biocontrol. And then we have to develop further for the application, and a lot of things should be done in this to make it available, can be really used for the biocontrol. But then also, actually, those knowledge in the metabolite of interest, you can use it for the biomarker. Even sometimes you can use it for the engineering of your line, yeah, or your cultivar. And then you can use it for the marker-assisted breeding. Yeah, you will get you can choose your varieties that have a good trait, and you can use it for the breeding. Okay, so this is, I just want to share first about the how we do or apply the metabolomics for my study. So this is one of the two study that's work in the tomato genotype, for tomato genotype for the root knot nematode. So here we already identify for cultivar, yeah, we got which one is resistant and susceptible using the bioassay that we apply with the nematode here. So we can find, we score it, okay, one is resistant, one is susceptible. Then we compare the metabolome using metabolomics. And then after we compare the metabolome, then we will have to do with the multivariate data analysis or chemometric analysis. Then normally we will get a sort of score plot that show how is your sample will be grouped. If you have this good separation. So of course you are also lucky there. You need a lucky here. Yeah. So in my study, in my case, it's really separate very well between the resistant and the susceptible. So then I want to know which metabolite will actually different between both. So then I go, here to what we call loading plot. In the loading plot, then we will identify several metabolites that may important in the resistant part here, the left side. Yeah, so we go to the left side and then try to identify, and we find it that the caffeic acid and glucose, the both alpha and beta is important for the resistant character of the tomato against the root knot nematodes. And here we try to, and then we do the quantitative analysis, and we found it in the GM2, which is the resistant cultivar, is indeed after the infection, the white one, yeah, after the infection, it's much higher compared with the other susceptible varieties. So then we think that we may work with this compound for further development for biocontrol. Yeah, and study here is using the rice ear bug. So in Gajah Mada University, we also have a group of research group. Yeah, we're working with pigmented rice. We are using the omics approach there, from the transcriptomics, genomics, and also metabolomics. And why? Because we know that pigmented rice is a healthy food rich with anthocyanin, and it's really good to be developed as a functional food. But the problem that the study on the cultivar, which price superior for this, is not really much. So that's why then we work with this. And especially the rice ear bug is still become a problem in the pigmented rice. So then we study about this using the NMR-based metabolomic approach, just the same like previous one. We first we have a collection like 40 pigmented rice in our group. Yeah, it's local pigmented rice from Indonesia. We try to collect it from everywhere in Indonesia, from Kalimantan, for Papua, and everywhere. Yeah. Then we do the bioassay. We decide which one is resistant, which one is susceptible. Then we compare it the metabolome, and we got that those compound seems important in the resistancy of pigmented rice against rice ear bug. So like here, for instance, you will see that the resistant one is quite separate very well with the susceptible one. So basically it will always the same like that step. Yeah. For instance, this is our maybe like 2 months ago. Yeah, we just received our publication just appeared in Plants. It's quite good. Also, journals. It's also working with tomato, but it's against bacterial wilt. Yeah, and we see here the same like the previous with the nematode, but now we are using the bacterial wilt as a treatment. And then we do the metabolomics, we do the NMR analysis in the leaves of metabolome, yeah, and then we do the chemometrics, or multivariate data analysis. And this, from the score plot, we found a good separation between the resistant one and the susceptible one. Then we check. Sometimes you can also not only working with the loading plot, you also can work with the VIP score here. That will give you an idea which metabolite can be important in this resistant character. We see here that in the resistant character, this is the leucine. This is the ppm of the compound. Yeah. So this is the leucine here, and this is the valine, which is this one is higher in the resistant, and this one is higher in the susceptible one. It's interesting because valine and leucine they are connected very well in the synthesis. They are both is known as the purified amino acid synthesis, and so we hypothesize that maybe the low concentration of valine in the resistant, because the metabolomics or the metabolism is prefer more to the leucine. So because that's why, and the leucine is higher because it's needed for the, remember that in some of you that work with the plant, maybe you know that there is a receptor known as NPS, LRR, leucine-rich lipid protein receptor, which this receptor usually will interact with your pathogen, and this will give you that will turn on the defense mechanism in the plant. So seems like what we think in the resistant, this NPS LRR is higher compared with the susceptible one. But of course, we are now still working in the transcriptomic level to prove this argument. Yeah. Then the other, okay. So those three is I want to give you the idea how we do with the metabolomics in my study. Then how actually the metabolomics can be done. So normally we divide into 5 flow. Yeah, the first is you have to do the experimental design, and then sample preparation, data acquisition, data processing, and statistical analysis. So it's always like this. It's only 5 part. It's not really difficult actually, but one of the most important is this part. The first part is, I'm sorry, maybe it's okay. The experimental design is very important because you have here to design or to take all the possible factors that will affect your metabolome. You try to identify all first, and then you design your research there. Because if this is poorly designed, so what you will have in this part, if you even have, maybe it will be misleading. So that's why you really take care of this. Yeah. First, you should put the strong research question, and then try to design your research carefully. How is that? For instance, in my study, I usually treat with my samples, yeah, with some of maybe like herbivore insect, maybe sometimes with bacteria, fungi, or virus. I have also PhD student working with virus now. Yeah, so yeah, you will put those treatment to your samples, right? So you have to make sure that all of the factor aside from your treatment should be all so the same. So when you get the response metabolism, you make sure it's just because of your treatment and not because of your other factors. Yeah. So how we do when we do the bioassay? So you have to make sure that I will just change this. Okay, to this one. Yeah, you have make sure that all of the plant receive the same treatment. So you have to identify all the factor and then make it all receive the same treatment. Yeah, the research design, you have to first make sure that you have to consider also the biological variation, because you work with the organism. So at least I use 5 replicates. That's the least. Cannot be less. Yeah. Then if you have, like here, you have 7 cultivars, so it's 35 samples already. And then if you will use this both, so we'll time 2, so it's become 17. And you know that the analytical tools is quite expensive, so that's why, of course, it sometimes become the hit, because metabolomics is a bit expensive. Yeah. So here, normally what I do is I have 2 set, yeah, 2 set of research here. First is I usually not infected. I don't put anything there. I let it grow normally, and the other one is the one that I will treat it. Whether I will treat with the herbivore insect or with pathogen, whatever it is that. But so there's always 2 here. Why? Because it depends to your cultivar or your species. Sometimes plant, it has the constitutive defense, but some they have the inductive defense. So if your plant known it has inductive defense, so I suggest you to compare more after the infection and before the infection, aside from the resistant and also the susceptible one. So you compare the resistant and susceptible varieties, and also you compare it with the before and after, because you want to see how's the response there. What is the compound which actually become activated after the infection, because it's inductive defense. But if there is no inductive defense, so I choose it this one, the non-infected one. So it will more look at the genetic background of the plants, yeah, and how it's different between one and the others, not because of that really the infection, but because of the background of the genetic, yeah. So after, and then here, like I said, that you have to identify all parameters that may affect the result aside from the treatment we will apply. So the important factor here, yeah, you have to first think what sample you will use. Yeah, you will choose the leaf, or you will choose the root, or you will just do in the level of tissue, or maybe a specific cell. It's really depend on to your sample. Maybe sometimes the latex, I have studied together with France, we work with the latex of Hevea, so we put the latex, yeah. So it's depend to your question also which one. So that's why I say that you really need different, you really need to define your research question very well. Then you also should think what analytical tools you will use. For instance, I use a lot with NMR, but sometimes I also use with GCMS. So the problem with NMR is the limit of detection. It's quite not sensitive, so it needs a lot of sample. So for instance, in the study of the virus, the bioassay is done in the seedling time. So it's really still tiny plant. So with this only one plant, I cannot detect with the NMR for sure. So that's why then I have to combine. Yeah. So the replication is not as organism, but then the replication is become pot, or maybe like polypeg. So one polypeg I have like 2 or 3 shoot. Yeah. So then it's one replicate, and then I have another polypeg with this. So that's why it's become, you have to think about this. Then also map the variation. You have to make sure about the diurnal variation, the microenvironment. You should make sure that all of your plant or sample receiving the same shading, humidity, fertilizer, and then also when you will do the collection, is it in the dry or rainy season? And then also make sure that they have similar treatment. Yeah. So then those some of the factors that you have to think about. Then how about the sample preparation? You have to think about the metabolites, that because the metabolite, it has a lot of different, yeah, in single organism you have different concentration. Some is very small, some is very huge, like sugar in a plant is always very abundant. And then the chemical behavior, one is polar, one is non-polar. Yeah, stability was stable in the hot, another is with heat, it's maybe destroyed. Yeah, so you have to think about it. And so for this, the first what you do is, of course, you have to stop first the metabolism. So when you collect the sample, you have to stop all the biological process during the collection by quenching. So that's why when I collect my sample, usually I bring liquid nitrogen. Yeah, because when I collect, I in the liquid nitrogen to stop all of the metabolic process. Yeah, just cooling and then work fast. Yeah, and then put it in the -80. Yeah, then we will work with this during when the extraction is needed. So usually the pre-extraction is like you will grind it and then remove the water content, because I work a lot with the NMR, which water has become a big problem. So I always remove the water content using the freeze drying. Yeah, and then do the extraction. The extraction is, we usually in metabolomics, we choose the simplest one. one solvent is better. The more simpler, yeah, the simpler one is the better one. Yeah. Why? Because you don't want to put a lot of your interview. Maybe because when you do a lot of step, you may interfere your metabolite. You may put the contamination there. So that's why one single solvent is the best actually. But sometimes you cannot do that. So some people they try to separate between the polar and non-polar. For instance, me and the latex, yeah, I have to separate between the non-polar and polar because of the latex. It's really non-polar, so I want to get the polar one of the compound, the polar compound. So I have to do this separation. And sometimes you also need to do the SPE, solid phase extraction, but those is actually not really favorable. Like I say that sometimes when you put a lot of step there, we afraid you will change your metabolite. Yeah. So in the metabolomics, we don't really do like that. Yeah. So single extraction is the best actually. Then you do the data acquisition. You can use NMR, you can use GCMS-MS, LCMS-MS, which one is available, that's do it with this. Yeah. So like I said, you have the sample and then you put this in the separation for the extraction. And then with the extraction, you separate it and then you do the detection. There are usually three tools that you use for the separation here. You can use the GC, you can use the LC, or sometimes the capillary electrophoresis. GC is good because it's reproducible, it's high reproducible, and then also have a very high resolution. But the problem here, you see that the range of the compound can be detected is quite narrow. Yeah, it's only for the primary metabolite there, yeah. And then for the LC, it's broader, yeah, the range, but the problem is the resolution is not really high, and then also it's not really reproducible. Also, that's also become the problem. And the CE, it's only applicable for the charts metabolite, so there is no very ideal one. So you have to choose, and you have to know that what. You have to think that, okay, this is my problem. In terms of the sensitivity, we know that NMR is the lowest. Yeah, you have to have a lot of samples there because they work on up to the millimolar. Yeah, GC is micromolar and some a bit nanomolar, but the LC is much better. It's up to the picomolar. And also here the metabolite detected, yeah. So of course LC is the highest, NMR is quite low because when you work with extract with a lot of metabolites, so you will have a lot of how can I say that, clouded signal and difficult for you to identify. So this is separate how to compare it between the NMR GCMS. Yeah, you can check it later. That the compound identification is quite difficult. That's the problem with the NMR, but the handling is very simple and fast. GCMS is relatively easier, but it's need you have to derivatize your sample at least 4 hours. You have to do with this, so it's a bit not really favorable for in this term, yeah, and it's also quite more expensive. Then you do the data processing. When you got the chromatogram, and then you do the packeting, then you do the data processing here. You try, you get the chromatogram, you do the baseline correction, peak detection, peak alignment, normalization. Of course, you have to do this, yeah, because you have a lot of signal with different concentration, yeah. So for sure, your sample, your variable will not be normal distribution, yeah, not normally distributed. You have to do the normalization and then also the convolution sometimes. Then you will go to the statistical analysis. So how about data processing? Normally you will get like around 100 to 200 metabolites. You can be recognized using the GCMS, detected using GCMS. LCMS, it depends your LC. If you have a very good LCMS, you can go up to 500 maybe. Yeah. For the NMR, it's actually the poorest one for the detection. Normally we can go up to 50. But then for the NMR, because of this, normally what we do is we don't identify the compound, but then what we do is we do the bucketing. So if we measure from 0 to 10 ppm, later I will show you. So what we do is we just do the bucketing, and then up to 200, up to, so normally the bucketing is the weight is around 0.4 ppm. So then after that you will get around 200 variables. Then you will use this for the metabolomics. You don't really go to the identification first, but you go with this bucketing first. Infrared is also the same. You will do the bucketing, and then it will be around 100 there variables you can get, because you have a lot of data. So that's why then you have to do with the multivariate data analysis or with the chemometrics. Yeah, here you have to do those chemometric analysis. I cannot really mention here, but if you want, any one of you, maybe later you can just approach me. I can show you how to do, because usually when I do this with my student, I take like 100 minutes just for the simple introduction. Then we go to the directly to the to practice it. Yeah, then you can do it normally. Yeah, after that, it's quite a simple one for the beginner one. Yeah, it's okay. If there is also a very good platform online there which is free, so you can use it very. It's quite easy there. But I'm sorry, I cannot do it here because the time limitation. Then after you do this one, sometimes also you need to do the spectra identification, because for the non-targeted you really need to do this. And there are a lot of platforms also now that you can use for the database for the spectra identification. You can use PubChem. I use a lot of the HMDB, sometimes the Mine, this one, or KHG. So there are a lot of databases that you can use. Then for the chemometric or multivariate analysis, you can use the unsupervised. This is the principal component analysis, or the supervised one, which is partialis, and then PLSDA or sparse PSCA. So those you can use it. But again, I cannot really mention it here. But later, if you interested on this, just approach me later. Yeah, okay. And the software that we need, there are some is really expensive one, like Simca and Unscramble X. It's you have to put a lot of money there. Though I have also one here, but then now I try to change it to the free one. This is the MetaboAnalyst, which is available online, free, and it's also powerful one. So we use a lot now with this, and also with the R. With the Mixomics package, you can use R, and it's also powerful one. Then, so what usually you get from those multivariate data analysis is you will get the score plot and loading plot. The score plot is showing you the distribution of your sample. For instance, I remember that I always compare between the resistant and the susceptible. Rentan is susceptible, sorry, here it's still in Indonesia, and then tahan is the resistant one. Normally, if you got a good model, then you will have that good separation. Yeah, for instance, this is the resistant is very well separated from the susceptible one. And then if you want to see or you want to check which metabolite is responsible for this, you go to the loading plot. Here in the loading plot, and then you can check which, for instance, this one is in the right side, right? Because this is the zero, sorry. I will show you here. This is the zero here, the zero line, yeah. So this is in the right side, this is the left side. So the right side here, this compound may be responsible for the resistant character. So you can check it, and then you get this. This normally the software will give you directly those metabolite and those charts. Yeah. Sometimes you also can go directly to the VIP, variable important in project, and then you will see some of compound which may responsible also important for the character that you choose, for the resistance. Yeah. So those usually we will work further for maybe like the development of maybe biocontrol, or for later for genetic engineering, and maybe you can just apply it just to see how is that will be affect to the physiological of your plant. Yeah. Then sometimes, if needed, you can also use the software to make the dendrogram and also the heat map of your compound. So it's really handy one. Yeah. Then this is I'm sorry, how many time that I still have? How many minutes that I still have? I don't know.
You still have about half an hour actually.
Oh, so it's quite a lot.
Until 10:20. 10:20,
Oh,
I think.
10:20. Okay. Now
Yeah.
I didn't really check it. Okay. Okay, so it's good. I will a bit slower than now.
Right.
This is the one of my first study after my PhD, the NMR metabolomic study on the pigmented rice resistant to Pyricularia grisea. So again, it's pigmented rice because we have this research group in there. Yeah. So what we do as plus it still become also a problem in the pigmented rice agriculture, because it's infects rice in all stage of growth and development. Yeah. So that's why we think that we should really get the good cultivar which resistant to plus. Yeah. So here we choose the metabolomics to understand how is the mechanism of the resistancy, and there is no known cultivar which resistant to plus now in the pigmented rice. So that's why we really think we should have it. Yeah. So this is what I forgot to mention also. When you do this, for instance, you have a collection of cultivar that you don't know how is the status of the resistancy. Yeah. So you should always have the control cultivar. So you should have known there is one or two which is known the resistancy status of that cultivar. For instance, here I use Asahan as the resistant cultivar. Susceptible cultivar is I use Kencana Bali. Both is actually the white cultivar, but I use it as a control because to make sure that your bioassay working well, you should have this control. Yeah. So then I treated this 7 cultivar using with the blast Funga Pyricularia grisea. Then, like I mentioned previously, that I have the control and the silk resistant and susceptible cultivar, and I have the unknown resistancy cultivar status here, like A to G. Yeah, and then I do the bioassay there to understand how is the status there between this cultivar. So I got there that using those bioassay, that's from the 7, this CIW and CAW, which is stand from Cempo Ireng wild type and Cempo Abang Wangen. This is the local cultivar from Java. Yeah, it's very very resistant. Yeah, you can see that the low bone here, the score of the bone here is the score. Yeah, this is the cultivar. The score is very low, so it's mean that you have a very low, like less than 1, so it's still considered as resistant. Then you also have you see here the positive control. It's nothing. They don't have 1 here. Then you see the susceptible one. Here is the IH. That stand from Ireng Imbira. It's also have very high score, which is around 4.5, almost 5. Yeah, which is considered as moderate. This is supposed to be moderate resistant. Yeah, moderate resistant or susceptible. But if you check here, the negative control is almost similar. So that's why then I always use the most resistant and the most susceptible one. So I compare between these 5 cultivar for the metabolomics. I use NMR. So this is normally how typically the NMR chromatogram that I got from those measurement from 0 ppm to 10, yeah, and then I try to identify some of the compound there. It's very difficult because a lot of overlapping signal. Yeah, you have a lot of time there. But sometimes when you do the 2D NMR, then it will help a lot actually. Yeah, so then I can identify several compounds there, and then I go, and then yeah, like I mentioned that usually when I do with this NMR, we don't pick one of the signal, but I do the bucketing. So the bucketing is just like you put how can I say that? Like you divide those area with the 0.4 ppm. So there are quite a lot of area here, and then you measure the area compared with the whole area, and then you get the number there. So that's why then it's become the variable. Yeah. Then after that you get those number. Then you go with the multivariate data analysis. You run it with the software. Don't ask me how is the algorithm of the PCA. I don't know because I'm just using also. Then when, for instance, you do the PCA, you do the PLSDA, there are several things that you have to consider there actually. But then after that, you will get the score plot. Remember that the score plot is showing you how is the distribution of your samples. And here, to get a good separation, to get a good model, like I mentioned, usually I use 5 replicates at least. Yeah, if you got 3, you do 3. It's not really good sometimes. So that's why I suggest you to have a lot of replicate there to make it the model become better. Yeah, and I'm lucky because I have quite a good separation. Here is the red is the and the blue one is the susceptible sample. Then I go to the loading plot. Yeah, here the loading plot. So all of the compound or the signal which is close the same direction with the resistance here, so it will may responsible for the resistant character, and if it's the same part in the susceptible part, so it's also may responsible for the susceptible one. So when we check a lot with this signal, we found it that the ferulic acid, GABA, and then choline is high in the resistance, and also the phenylalanine and tyrosine there is also high, and the susceptible cultivar it's higher in sugar. So it's interesting for me. Then we are still now working with whether this ferulic can be proposed later further because it's available commercially. Yeah. So we are thinking to use it further, maybe for application or additional of the biocontrol. Yeah. So that's why it seems like it's good candidate. Yeah. So those the way we usually do. And because it's pigmented rice is different from white rice, remember that it has high anthocyanin, and anthocyanin is produced from the sikimate pathway. Yeah, remember this. And the sikimate is actually producing also those aromatic amino acid, which was detected higher in the resistant. So we think that maybe that make it the pigmented rice also. How can I say that? If you look at the previous, it's many of the pigmented rice is not really high actually. It's like high susceptible. It's more to the resistant and the moderate resistant. Maybe because of this activated metabolism there that may support the resistant character in the pigmented rice. Yeah. So we are still working with this. We are looking possibility like to do the engineering character in this pigmented rice with this metabolic pathway. And those, so up to the end, yeah, the take-home message here that the metabolism is a powerful tools to study plant resistance and mechanism. And then remember that you should have a strong research question so you can choose which sample you will use, and then do first try to identify all of the factors that may affect your plant's metabolome. Then and design your research very well. Yeah, this is very important and crucial for the successful of metabolomic study. And then after that, we have to know that metabolomics is still the early step of the biocontrol study. You still need a lot of things that you have to do the bioassay again, and then to check whether those really important for the work for the biocontrol, and then also to develop into the applicable biocontrol is still need a further step for sure. And then again, like I mentioned in the previous, need several skills. Metabolomics. You need maybe like me. I'm not really good. I'm not the master in chemistry. But I know a bit of chemistry. Maybe I'm good in biology, and I know a bit also about bioinformatics. So it's work. But if you think that you are not really master in those all, just do the collaboration. So collaboration is one of the best solution here. Okay, so then if the previous one you see that metabolomics is still blurred, I hope after this you have quite better picture about metabolomics. And thank you. And I will open further discussion after this. Thank you, Mas. I'll return it to the moderator.
All right. Okay. Thank you, Dr. Nuning. That is a very insightful lectures. Actually, I never thought that metabolics would be that deep actually and encompass such a wide variety of subjects, even involving data processing. And what surprised me, it's actually involved also signal processing.
Exactly.
Okay. But first, before we are going to the Q&A session, we would like to take a break first for the participant to process all the information you have just delivered to them.
Okay.
Please enjoy this break video first. Okay. Okay, that should be enough mind break. No, that's a bad term. I think it should be a nice break to refresh your brain after this a very huge subject lectures. So I invite all participants to raise your hand if you have any question that you would like to directly ask to Dr. Nuning over here. You can also put your question into the chat room or into the link available on the chat room there at slide.com/vss8. So, is there anyone who want to ask Dr. Ningrum directly? Please raise your hand. Okay, I think we'll start with AHA slide first. So over here we have Arif. Arif, are you here? Arif J, I think that Arif Juliari. Hello? Arif? Can you hear me? Okay, I'll just read his question. Thank you for the great explanation, Bu Nuringkes. What kind of data processing or analysis software that commonly used in metabolomics? So let's suggest the free one for us. I think you have actually answered that question, but I think you can elaborate further on that.
Yes.
Please, Dr. Nuning.
Thank you for the question, Mas Arif. Yeah. So like I've mentioned previously that I use a lot with Metabu analysis. Yeah, you can try to check it. No, also there is metabuanalysis.ca. Yeah. So then you will see there are a lot of platform free. It's all free there, and then it's really powerful as well, and also quite easy. And it's there you can do the PCA, you can do the PLSDA, you can do all of the chemometric analysis. You can also check for the biomarker analysis there. And I think it's really powerful when it's really free. So it's free there. So the important thing is how can I say that? The result of the graphic is quite good as well. So just use this. But you should remember if you have because this is not only for metabolomics, yeah. Actually, you can also use for other, if you have a lot of variable, then you want to do the multivariate data analysis, you can use that. But the most important, you can submit that there online, is you don't put in the Excel. If you use the Excel, you just use the tab delimited, yeah, and or maybe the text file. So you have to save as your Excel to the tab delimited CSV, yeah, or maybe to the text file. Then you can submit that. You can work with this. So actually, it's very good and very easy. Usually, I introduce this to my student as well in the bioinformatics study, and they can do it after that for maybe 2 times 2 hours. Yeah, we can learn, and then they practice how to do, and then you can do that. It's easy. Yeah.
One of your most recommended is Metabo Analysis, right?
Yeah, metaboanalysis.ca. Yeah, you can go there. Yeah.
Right. So a web-based software.
Yes.
That is actually very interesting. I think I should try that one. I'm not exactly work in metabolomics, but I'm sure interested in learning one. So I think I should try it. Right. The next one is Vinny. I know Vinny is here. I've checked. Vinny, can you hear me? Vinny is actually my junior. She is currently she was my junior when we are in Gajah Mada University. So both of us actually graduated from Gajah Mada University,
Okay.
chemistry department. Vini, can you hear me? Hello, Vini.
Yes.
So maybe you can ask Dr. Noning Eng directly.
Yeah. Well, thank you for a great presentation. Simple. It's just about your comment about every field in chemistry, about how machine metabolomic analysis. Since like a few weeks ago, I attended a webinar which the research group developing a software called MS Query for spectrochemistry, which which use machine learning for analysis. And I just like to ask you what your comments about how the trends in Indonesia. Thank you.
Yeah. Thank you very much for the interesting question. Yeah, I think because I think this is one of the development after the basic metabolomics. Then actually after if you have a lot of data already and you do it with the same topics and then use the same parameters or the same activity that you want, and then you have quite a lot of data and then actually you can do you can work with the machine learning. I'm not really I'm not a master in machine learning because this is not my background, yeah. But I think it's very good, yeah, as long as you make sure that you have a lot of when you develop the machine learning, you have this background as the data is quite already validated very well, and then yeah, it can be used for that. It can be further developed into machine learning. I work a lot with Pak Wisnu actually also. We are working with for the agar, because I also working with Agarwood, yeah, Gaharu, for the drug discovery, and we are now still working with this, and we use his machine learning, yeah, to study the possibility for using this for the what is that? Not really COVID, but it's more for the invalidation, yeah. Yeah, so it's good, especially for the drug discovery, I think, yes. Yeah.
Okay, Vini, perhaps you have further question regarding that?
No, thank you.
Okay. So actually involving machine learning, yeah, actually when I look at data, when you are talking about principal component analysis, I think machine learning would work just fine with this thing. Of course, we
But it's
working with
actually
like
yeah,
big data, right?
yeah. You should have a lot of data first to develop your machine learning. Yeah.
Yeah. But of course, when we are working with big data, we spend a lot of them. Sometimes just verifying whether the data we have is actually correct, right?
Yes, exactly.
So to be honest, I usually when we're working with statistics, most of the time I actually said that it's not difficult, it's just annoying. Because we have to check and recheck the data again and again.
Yes.
The next one is from Nilofar. All right, Nilofar, can you hear me? I think Nilofar is here. She's usually here. Yeah, I can see her here. Hello, Nilofar?
I'm here,
Right.
sister.
Would you like to ask the question directly? Perhaps you can open your mic. Okay, she said that she has internet problems. Right. So here is her question. Why is lyophil? Is it lyophilization? Is that correct? Or is it lipophilization?
No,
Utilization, right?
lyophilization. Yeah, that's
Required
right.
for the chloroform fraction.
Yeah.
Are there any volatile chemicals in this fraction?
Okay.
Okay.
Maybe the way I it's not really correct. Maybe the way I explain it's not really clear. Yeah. I mean here the lyophilization is I did it before the extraction. Yeah. So normally after I grind the sample, you remember that usually I put it in the liquid nitrogen first because I want to quench to do the quenching, yeah, to stop all of the metabolite process, the metabolic process there. Then after that I grind with the liquid nitrogen as well, and then I send it for the lyophilization because I want to remove the water. So the lyophilization is more for before the extraction. But then after I got the dry samples, then I do the extraction. Yes. So it's not I never do the fraction the lyophilization with the for the chloroform and also for the other. So because directly I normally just use it after the dry samples, then I extract directly using the NMR solvent. Yeah, because NMR is very simple. So when you have the dry samples, then you just put your solvent, you sonicate to put in the sonicator, and then you spin it down, and then you got the supernatant, and then you send it. You put it in the NMR. That's it. You got the data. Yeah. So it's really simple, very simple step for the NMR. Yeah.
Thank you. So it's basically a technical problem, isn't it?
Yeah.
Yeah. All right. Okay. Are there anyone else who want to ask question? If you have any question, you can raise your hand, and we can let you open your mic, unmute your mic, and you can ask directly, or you can also put your question in the AHA slide. The link is given on the chat room. Or the easiest one, you can just put your question in the chat room and we'll put it in the AHA slide. Right. Okay. The next one, this one is one from one of the committee, I believe, but she's currently going to the clinic near her house, so I'm not sure she can ask the question directly.
I think she's there.
Her question is, how did you do the interpretation for
Hello,
NMR
Ibu
spectra?
Amel.
Oh, she's here.
Yeah.
Please, Bu Amel.
Okay, Dr. Nuning, before I ask the question, I will say thank you so much for accept our invitation in the virtual summer school. Okay, Dr. Nuning.
Thank you very much also for the opportunity to share it here.
Yeah. For me, this is a new insight, especially most of the participants from chemistry major, but all of them from the biology, and you especially from biology. I think it's very nice to share another major, research. Okay, doctor. I would like to ask about NMR spectra because before the speakers explain about how to interpret the pure compound from NMR. But in this
Okay.
metabolomics, the samples is extract, right? So there's a contain many
Yes, it's
secondary metabolites. And what did you do how did you do the interpretation for the NMR? And especially what kind of the 2D NMR experiment that you use for the metabolomics? Thank you, Dr. Nuning.
yeah. You're welcome. It's very good question, actually. Yeah, that's become the most problem, especially when you use the NMR spectra for metabolomics, because you will have so many signal there. It's not like when you have a pure compound, so then you can do do the correlation, whatever. So you are really sure that all of those is really important, or that's your really sample, your really compound. Yeah, but in the metabolomics, that's why sometimes it's difficult because those overlapping signal. So what we do is usually we also do the 2D, especially J-resolve. Yeah, we do the J-resolve. There you will get the coupling constant. Yeah, and then if it's a double, if it's so overlapping signal, when you do the J-resolve, then you will see that how many, what is the actual signal there. For instance, it seems like it's doublet, maybe a singlet, or maybe a triplet. Sometimes it's difficult, right? So that's why do the J-resolve there. And when you do the J-resolve, you can get exactly what is that there. Yeah. That's the most important is first you do the J-resolve. So normally I only do one of your, because normally it's one species, right? So it doesn't matter. You will just do one of your sample, put it in the J-resolve. Then you can use this for all of your sample. And also you can use the COSI there, yeah, because it's helped a lot, especially when you want to make sure in the aromatic reagent, for instance, whether this is coupling one or, yeah, the signal is really correlated. Sometimes you, like in the ferulic acid, yeah, you have to really make sure whether this is really that sample. So especially for the one which is appear in the aromatic, I use also a lot with the COSI. Yeah, that's the most that I use, J-resolve and COSI. And then, of course, we also have in our lab, we also already run a lot of a pure compound. Yeah, we have quite like a database of NMR spectra that consists of some of the pure compound that of our interest. So then we can compare it directly after that. We put those put it together, and then we will see. Okay, it seems like for the beginner one, this is really help. Yeah, so you will just compare it then with the standard data that we have. Yeah, like that, Ms. Bu Amelinda. Will it help?
Thank you so much, Doctor. So I think it's a lot of work,
Yes.
especially for the isolation, the secondary metabolites that we curious about it. I think it's nice, the answer. Thank you, Doctor.
Welcome.
Okay, that was from Bu Amelinda. I actually have my own question for this actually. So in the research flow, you have like 5 steps, including acquisition process, experimental design, sample preparation, up to statistical analysis.
Yeah.
So you actually mentioned univariate and multivariate analysis over there. So what kind of analysis did you use other than PCA? Of course, we can see it clearly, the one principal component analysis. Other than PCA, what kind of other statistical analysis did you use in this research?
The first is, of course, I always use the multivariate data analysis. The chemometric is always there first. But then after that, you will try to quantify, right, the signal. You want to quantify each of the compounds there. So then you want to make sure whether this, for instance, from the loading plot, you found there that, okay, it seems like this is glutamate, this is ferulic acid or whatever, sometimes chlorogenic acid, yeah, there. So then you want to make sure whether this is really a significantly different or not. So then after that, you will do the univariate. Yeah, you will do, like, maybe depend to your research design, whether you will do the ANOVA or after that, or 2D ANOVA. Yeah, something 2-way ANOVA, yeah, something like that. So, or sometimes already tests. Yeah, it depends to your design. Yeah, but then normally you do the multivariate first, and then you pinpoint the metabolite there. Then after that, you quantify the compound, the concentration, and then those concentration you will need to do with the ANOVA or the univariate one. Yeah, or sometimes the test, or 2-way. Is it
that actually
answering?
So it's basically depends on what you are trying to do, isn't it?
Yes.
So what
So
actually
your question, yeah, your
you want
question
from
is
your
really,
research.
yeah.
Yeah. All right. Okay. Of course, once again, I invite the participants to raise your hand if you have any question you want to ask to Dr. Nuning directly. You can also put your question in the chat room, and in the AHA slide, the link is given in the chat room. So next, this one is from, I don't know who's this is. We have an anonymous question over here. So how did you do the preparation sample? You have, because you mentioned before that metabolomic especially are very sensitive to environmental response. Yeah. So how exactly did you do that?
Okay. I don't know. I mean, I'm not really sure with the question. If you mention about the environmental response, so here it's more working with the design, right? Because you have to make sure that all of the variable or factors that may affect your plant samples there is controlled. Yeah. For instance, like humidity, the shading, or whatever is that maybe the age of the plant should be the same, right? So everything. So you really have to take care all. You make lists of that all. Yeah. Then after that, you have to make sure that those variable is controlled well. And then after that, you do the treatment. And then after the end of your bioassay, then you will do the collection, right? The collection is normally, sometimes it's a bit far from your lab. For instance, when I do the HEVIA study, I have to do to Sumbawa, Palembang. It is really far from Yogyakarta. So then after that, I have to travel to go to Yogyakarta to bring it to my lab. Yeah. So what I did is I have to what's that, dry ice. Yeah. So I bring dry ice with them in a flight, yeah, from Jogja to Palembang, because I cannot find dry ice there. Maybe in the city is there, but in Sumbawa there is no dry ice. So that's why I bring it by myself. So it's really complicated. So it's really depend on your situation there. So in the collection, you have to make sure that you do the quenching. Yeah, you put it in the liquid nitrogen directly to stop everything there. So because liquid nitrogen is like minus 100, 120, yeah, I think minus 120 degree. Yeah, so it's all stopped there. Yeah, the biological process, and then you have to make sure that you put always in a cold condition. Yeah, that's why I bring this dry ice from Jogja to Palembang. Remember that. And sometimes
keep it fresh. Keep it fresh.
Yeah,
keep
sometimes I also just bring liquid nitrogen if it's within still in the distance of what's that, in Jogja or to Jawa Tengah, to Central Java. So I just bring my tank, liquid tank, and then I bring it to the lab. So that's the most important in the collection. Yeah, put everything in the cold, always in the cold. Yeah, should be remembered that. Then after that, yeah, we do because I use a lot with NMR, so that's why I always put it in the the freeze dryer, yeah, to lyophilize. Yeah, and when it's already dry, then I do the extraction. That's it. So but the most difficult part is not the chemical reaction, not the extraction, but when you really keep everything in cold, and then you have to do the research design, you make sure everything is the correct one, that the sample, that's the most problem in metabolomic. Yeah.
So basically, the sample preparation step is one of
the crucial step is really the research design. Yeah.
Yeah. So basically, you have to keep your sample as fresh as possible. I think would be, yeah. And then if you don't know other variables, try to make those variables the same across your samples, so it would be in control. Therefore, eliminate as much as possible the effect of the environment.
Yes,
that is actually very simple in principle, but I bet it would be very difficult in practice, especially with working, keeping the sample fresh. I mean, if from Central Java to Yogyakarta, I think that would be manageable. I mean, just like less than 200 kilometers, right? But from Palembang, I'm not sure you can keep it fresh for that long.
Finally, we did it, and we put dry ice. I put a lot of towel there and whatever. It's really crazy, but we can do it. Yeah, finally.
Yeah, it would be very difficult. Well, ideally, you would like when you have the sample, you just directly measure it on the spot, right? But that's just the ideal. The real world doesn't work like that. All right. The next question is, what should be preferred in sample preparation if we do not have the instrument? I think you have answered this indirectly.
You send for the analysis. Okay. Yeah. Yeah, I did it also, this one. Yeah. For instance, like for the GCMS. Because we have a lot of GCMS in Gajah Mada, but the problem is not as what I want. So when I do the GCMS MS metabolomic, so I have a collaboration with Saclay Institute in France, and also CIRAT in France. So there, what I did is I put it in the lyophilized condition first. So that's why I dried all of my samples in the freeze dry, and then when it's already dry in the lyophilized, so basically, actually, it's safe. You send it in the normal condition, without you don't need the ice condition anymore, because it's already dry. So then it's the time. So usually I send it when it's already lyophilized. So when like that, so you don't need any cold condition anymore. Yeah.
I see. So basically, just use freeze dry or dry ice or liquid nitrogen. Just keep it very cool. Okay. So let me ask the participant first. So participants, any question from the participants? All right. Well, the next question is, do you use specific software for data processing?
Yeah.
I think we have answered that question before. Your recommended one is Metaboloanalysis.
Yeah, but I use another also. Yeah, I use Simca also, which is expensive. It's around 30 million for 10 user only. It is really expensive one. And then I also study R, yeah. So I can use also R for the multivariate using the Mixomics package, yeah. And then I also use Metabo Analysis, but the easiest one is Metabo Analysis. Yeah, I can recommend it.
Okay, so Metabo Analysis. I really have to try that software. Okay, how many replication sample that used in metabolomic research?
Mm-hmm.
I'm not sure.
Yeah. Sometimes when thank you. Let me. Usually I use 5. Yeah, that's the list. So normally we have 5 replication, and sometimes 8 if it's really needed. Yeah, it's depend to the condition, but 5 usually it's normal. If I grow it in the quite how can I say, similar condition, so I use really 5. It's enough. But then if you use if you like the hevia, because hevia is a big plant, right? So it's really already there for a long time. It's in the field, so you will need more. So like 8 is a good enough. Yeah, because the environmental factor you cannot really the more you cannot control your environmental factor, so you need more replication for sure.
Right, so basically just the minimum is 5, but just get as much as you can, right? Basically.
Yeah, but it will be very, very expensive.
Yeah. Right. This is actually this is actually I think because I'm not really used to these topics, but is there any metabolomics study on from animals or non-plants organism?
Human. The first actually metabolomics is applied in the health. They call it metabonomic. Yeah. It's more using the blood, it's the first. They are even more. Previous one is more in the human, apply in the human actually for the biomarker. Yeah, but then the plan is a bit 2 or 3 years later after that. Yeah.
I see. Okay, I think that question is answered. So, how to wait? I missed it. Thanks for the presentation. Can you explain how to use multivariate analysis in the metabolomics example?
Do we have time for this? I don't know.
This one is pretty technical.
Yeah. If we have time, a lot of time actually.
Perhaps you can give us one example, the simplest example maybe.
Cannot be because it has to be demonstrated. Yeah. But at least, how can I say that? Okay, first, when you have the data, the set of your data, yeah, then first you have to do the normalization actually. So you have to normalize your sample. That usually I teach my student how to normalize, whether you will use the Pareto, you will use the CIFI or whatever there to make it your samples distribution is size, that one, the normal one. Then after that you do also the centering of your data, and then after that you go to the multivariate. When you do the multivariate, it's basically just click, yeah, because it's a software, you just click there. But it is important that you have to normalize first the sample. Yeah, and then you center the sample, you do the those are, because I cannot really do here because it's I cannot really explain it because you have to show actually there. Yeah, when you show, so if you have time, so maybe I can show actually maybe some of my, I don't know, if you like, I can show it a bit maybe from my.
Yes, please. I would like to see some of your examples.
Let me check. Wait a minute. Wait, I will find my lecture in bioinformatics. Wait, let's see. Okay, this one. Okay, okay. So I will just show to you. Informatic to support meta. Oh, this one. Okay. Yes. Can you see this?
Yes, I can see it.
Okay. So let me show you how is the look like, the tableau analysis look like. Now it's not really like this, but it's basically when you go to the metaboanalysis.ca, you will see this, you choose this statistical analysis. Now it's the 5 version is not like this, but basically just try to find the statistic analysis there and then click this, yeah. And then when you click, you will get like this. Now, then here you have to upload the one which is in the CSV. Yeah, so you have to make sure when you have the Excel, yeah, you save as to make it into CSV. Yeah, then you uploaded this. For me, because it is TCM, it was TCMS, the one that I use here. So I choose here the spec intensity. But if I use with the NMR data, I use the spectral bins. If it's really a concentration of samples, so then I use the concentration. But here, because I use the TCMS, it's more to the peak intensity. So that's why this is wrong. This should be the peak intensity. It's for the TCMS. Then I do the submit here. Then you will see, okay, and then when everything is correct, so there is no error, you can do the skip there. And then you click the proceed one, you put the none, choose the none here for the, because my sample is less than 500, so 5,000, so around 200 variables. So I usually just use none, and then here. So this is the look how you will normalize there. So when you do the you try to normalize your sample. First, you have to look first how your sample look like. For instance, you put everything is in none, and then you normalize. You will see here that, like here, before normalize, let me put here the lesser, yeah, here. So we see that the sample is not normal distribution, right? So what you do, try to do the, here, try to check what I do the scaling first. I use whether the auto scaling or Pareto scaling, and then do the normalize again. Check how is that compare between before the normalization and after normalization. Then if you got at least, a bit of quite good data already, the normalized data, so then you can proceed. Yeah. So this is really trial and error. I cannot say you have to do this one, but try those. Normally I do with this first, and then I rarely do with the log. But if it's really, this log is, if you find that your sample is really small concentration and there's huge, very huge concentration, then you really need to do the log transformation. But if not, so don't do this, just do this scaling, and maybe sometimes you can do with this normalization, like using the sum or median. Yeah. When you got already quite good normal condition, then do the next step. You click next, proceed, yeah, and then you will find like this. Then you can just choose PCA. First, you have to do the PCA, because PCA is unsupervised. What does unsupervised mean? That you don't put any information to the software. So the software will work with your sample, with your data, without any information. If it's really separate, so the separation is really based on the character of your data, not because you put any information there. Yeah. So usually I do with the PCA first, and then if it's good, I can further goes to the supervise. The supervise mean that you put as information in your software that your sample is actually consists of 2 different or 3 different things there. For instance, like me, usually it consists of resistant and susceptible one. So I put those information to the software, so the software will consider that the data is consists of 2 data. Yeah, so that's why you have first to do with the PCA first. Yeah, so that's it. So then you can get the principal component, you get the script plot, and then you get the score plot there. So those are the way how we do the in a glance, yeah, how we do the multivariate data analysis. And you can click and check it, how to do this, about how it will appears. And then if you have any question, then you can also contact me. Don't worry. I think I didn't put my email, yeah, but you can contact me in the tririni@ukmacad. So just feel free. I will try to to explain. That's it. Yeah.
Okay.
That's quite easy.
Thank you, Dr. Mling. That is actually very short demonstration, but actually very clear for such a short demonstration. So thank you very much. Okay, I think we still have question. So here is another one. How to freeze dry the sample? I mean, I think what he or she mean is there any extra preparation when you are freeze drying the sample?
Yeah, for sure. Normally I grind first. Yeah, I grind with the liquid nitrogen. Yeah, so it's become powder. Yeah, then you put in the vial. Yeah, or in the flacon. Yeah, tube, and then you put it in the freezer first. So it's better you also make it your sample freeze first, and then you put it inside the freeze dryer, because it will not really make your vacuum in the freeze dryer have to work very hard. Yeah, so it's better you freeze it put it in the freezer first. Yeah, after that. Yeah.
Okay. Next, what is the best instrument for metabolomics? I think like NMR
for the analytical tools. Yeah,
infrared,
maybe. Yeah.
Or
yeah. The best, I cannot say what is the best, because each of the tools actually have a advantage and disadvantage. Yeah. For instance, GCMS, MS, it's good, but you only can get primary metabolites, because when you do the what's that? The derivatization, yeah, you do the derivatization, then always mostly you will get is the primary metabolite. So sometimes when you don't want, if you want to get the secondary metabolite, you cannot really use with this GCMS. And then how about LCMS? If you have it, so it's good as well, but it's quite low reproducibility. And it's expensive, yeah. So if you have it, it's good. So that's why there's not really mentioned that what is the best. The best is what you can get. Yeah, I'm working a lot with NMR, because in UKM I have that platform. It's quite a good one. It's simple, so that's why I use a lot with this. Yeah, sometimes I use the CMS because I have a collaboration there. LCMS up to now, I'm not. I have also collaboration with the Warwick University in England, but it's how can I say? Yeah, when you have a lot of data, it's also difficult for you to interpret because when you have the LCMS, MS, so you will have like 300 metabolites there, you know. So the more you have, so the more difficult also you have to when you want to synthesize which one is the most important metabolite. So that's why it's depend what you have actually, I think.
So basically, depend on what you have and what you want to use it for.
Yes.
All right. So there is no best instrument, just what you have. Okay. I think this the next one had miss a word there. How to identify volatile compounds, because our lab does not have SPME. I think it's a solid phase micro extraction. Can look at extraction used to perform the extraction.
Yeah, and normally I use the more to the I use the not volatile compounds actually. So especially in the metabolomics, I use almost all with the normal liquid extraction. Yeah, because we are care more with the polar compounds, because later, especially if you want, especially for biocontrol also, you want to spray it, you want to use, you know, so yeah, normally we use with the polar compound. So that's for the volatile, I'm not really sure, because CCMS here when you do the metabolomics, you have to derivatize everything. So you also find the primary metabolites. So I cannot really answer this question. I don't know, but because I normally use with the liquid extraction, that's it. And in the metabolomics, we care more with those compound. With the volatile, maybe if you do the profiling with GC, it's also possible. Yeah, but then you don't do the derivatization. You just directly with the maybe with the how can I say that? I don't. With the gas, I don't know. With there is some equipment with this. I'm not really familiar also with the GC, and then you have those like a cup there, and then you will just directly measure those volatile. I don't know what is the name of this. I'm not really familiar as well, but maybe that's possible. Yeah.
I see. So this is the next one is the last question. Except for if the the participant have any other question, perhaps before we are moving to the last question here from participant. Putri Fatmawati, I see you are opening your mic. Do you have any question?
Yeah, sure. No, it's just now.
Hello? Putri Fatmawati, can you hear me? Okay, I think she can hear us. So this one is the last question, I think, from the participants. It's just that we don't know which participant this one is. So I want to ask, what if we don't know the resistant susceptible variant of the plants? For the comparison, can we just compare it with the plants without any treatment?
Yeah, so it really depends on your question. Yeah, what is your question? Is your question is like, there is another study in agarwood, my study, that we compare the metabolic profile of agarwood in different location, for instance, because I want to do the standardization of agarwood. So here I don't do any treatment. I just collect the sample from that the one from this field with another field. Yeah, so it's possible. So like what I said, it's depend to your research question. What is your research question and what do you want to answer it? If you like what I did, because my study is more for trying to find the metabolite which is responsible for the resistancy. So that's why I do the comparison of resistant and susceptible. But then if my question is more to the for instance, there is one of my student now, PhD student, also working with shallot. Yeah, she wants to know the difference between. There is one cultivar which is really red, the other is not really red. Shallot. Yeah. Then we want to know what's actually different between this red shallot with the not really red one. Yeah. So we don't do any treatment there. We just grow and then we collect it. Yeah. So it really depends on your question.
Oh, just a quick question regarding the red and not so red shallot there. How do we define red and not so red over there? Did we just use like some kind of the intensity of the redness or something like that?
There is charts, actually color charts from the Royal Horticulture company, research, something like that. Yeah, RHC, because she is from my PhD student is from agriculture. Yeah. So she's using this color chart from the Royal Horticulture. There is some degree of red of those shallot or whatever. So she can use that one to group the shallot. Yeah.
I see. That is actually that is one of the strangest one, I think, because I don't think that redness can be quantified. the best what I can think of when we're quantifying red or blue or color, something like that, we just like use the RGB scale, right? This one is so red. The red is like 252, something like that.
Yeah, some people they use the RGB, but for horticulture, for agriculture, they have this character. I don't know, it's like the charts for that one. Yeah.
Okay. All right. So thank you, Dr. Nuing. So I think that is the end of the discussion for this session. So I would like to extend our gratitude to you for thank you very much for participating in this virtual summer school program. This is a certificate from us to you.
Thank you very
as
much.
a speaker. So we will send it to your email directly.
Okay.
Hopefully we can send it as soon as possible. So once again, thank you very much. Before I think you will have some other things to do after this. So before that, perhaps you have some closing statement.
Well, thank you very much for everything, for inviting me also to take part of this virtual summer school. It's good to share here, and also I hope that more and more people is actually working with metabolomics. So we have for Indonesian, we have those association, yeah. Later maybe I will send to Bu Amelinda. I don't know, Bu Amelinda is already there or not, but feel free if you like, you can join, because later I think you will also have Pak Raffi, right? Raffi is one also my colleagues from IPB there. We are together working for the metabolomics in Indonesia. So if you like, you can also join there. You can always have a contact with us in that association, and then we can learn together for that. Okay.
All right. Thank you, Bu Nuning, for your great lectures. If you are if you have any other activities after this, you can leave the room. You're welcome to join us after this, because after this we have a quiz. So
maybe we have a photo session
after
before
this we have
Ibu
quiz
Nuning.
for all the participants.
I heard there was something. I don't know.
Excuse me, Ibu Nuning. My name is
Yeah.
Yanisa. Maybe we have any photo session.
All right.
Photo session. Okay.
We're going to take some pictures.
Okay,
Right.
okay. No problem.
Yeah.
Yes, sure.
Okay, Bu Nuning, thank you very much for joining with us. But before we close the session, but let me have a photo session first.
Sure.
So everyone who has any trouble with your connection, please open your video so we can take your picture. Okay, because there is 123 participants, Ibu Nuning, so maybe it will take quite a long time. Okay, thank you very much. So in 1, 2, 3, I will take a picture. 1, 2, 3. Next slide. 1, 2, 3. Okay. Next slide. 1, 2, 3. Okay. Next slide. 1, 2, 3. The last slide. 1, 2, 3. Okay. Okay, Ibu Tririni, thank you very much for joining
You're
with us.
welcome.
Yes,
Thank you, Ibu.
Thank you. So bye. If you have any more questions, just contact me. Thank you. I will leave. Yeah. Assalamualaikum.
Thank you very much. Waalaikumsalam.
Thank you, Nin.
All right, everyone. We have just finished the first lecture for today. First, I would like to ask the participants on how on your what do you think of the lecture for this first lecture? Let's see, who should I ask? We have a lot of people here. Let's start with Akib Talib. Can you hear me?
Yes, I hear you.
Yeah. So, what do you think about the lecture that have just been given?
Yes, it is pretty good and much knowledgeable and also a good lecture that presented earlier. And I also got much knowledge about metabololism, and it is a very good lecture.
Okay. Thank you. Okay. Next, who should I ask next? Let's ask Dea. Can you hear me, Dea? Dea Dimitri. That did I pronounce that correctly? Dea, can you hear me? Hello? I don't think she can hear me. So let's ask someone else. Let's see. Kanti, can you hear me? This one is one of our students, actually. Kanti Pawaningtyas, can you hear me?
Yes, I can hear you, sir.
Yeah. So, what do you think of the lectures that have just been delivered?
I think the lecture today is very insightful for me. Yes, sir.
Is that it? All right, I think that should be enough. Okay, I think that should be enough for the lectures, what the participant thinks about the lectures. I think we can move to the quiz now. Is the quiz ready? Okay.
Yes.
Yeah, the quiz is ready. So let me copy the link first. There you go. Please join the quiz. All right. We are still waiting for the players to join on the quiz. We have 8 players, 9, 12. All right, we almost to 50 players now. All right, we have 50 and still climbing. All right, we have 66 players. Whose screen is this? Right, this is from Bufidia. For those who don't remember, Bufidia is our first MC. All right, 67. Come on, we need more participant here. So there are 116 in this Zoom meeting. So I expect at least 90 of. All right. Is it still increasing? Why does it decrease? Come on. Let's see. I don't see Fini. Where is Fini? Right, I think we should wait some more. There you go. We have some more additional participants. Please join the quiz through the link given in the chat room. $74. Come on, come on. we'll still have some room. There are 85 that is live, but only 76 player. Where is the other? Where is the others? All right, 76 players. Okay, should we start now, Bu Vidya?
Yeah, if no more participants who wants to join the quiz, we can start the quiz. Because if you also have trouble with your connection, we will provide you the link maybe after the summer school through the Google Classroom or the Telegram. So.
Okay, I think we can start the quiz right now. Bu Vidya, please give us the countdown.
All right, I'm counting 3, 2, 1. Let's start the quiz.
All right, which one is the most sensitive to environmental response? Which one is it? If you are paying attention, you should know which one is the answer. I'm not going to tell you, but you can guess. Time's up. And the answer is metabolomics. All right. Most of the participants correctly answered it. All right. Let's go to the next question. First, Fan Tian. I hope I don't mispronounce your name, Fan Tian, with 89 points, and Angel gonna get right behind him. Next, which one is not correct about metabolomics? Quantitative, involving high molecular weights, include indigenous and exogenous molecules, a global identification for biological sample. All right, which one is it? Okay, the answer is B. All right, let's see. Is there any change to the leaderboard? Oh, now Farah is leading with 157 points. Okay, we have Dian in the second place. Which Dian is this? I think we have two Dian in here. Okay, let's go to the next question. The next question is, the following are specific purpose for targeted metabolomics except validation, identification, measurement of defined groups, require accuracy, high throughput, and reliability. Which one? Okay, the answer is chemical identification. Okay. Next is, Lee Tan-Hui is on top with Akib Talib right at his tail with 203, the difference of 5 points. Lisma is in the third place with 163 points. Let's see. Let's go to the next question. the main problem with untargeted approach is it identification, instrumentation, sample condition, or physiological state? Which one is it? Time is up, and the answer is chemical identification, which is surprising because almost same number of people actually answer sample condition, which is interesting. Okay, let's see how is the leaderboard change right now. Oh, Lee Tanfi is still on the top, but Akib Talib is closing in, and the difference is now just 3 points, while Saman is chasing from behind. Okay, let's go to the next question. Not an approach to investigate metabolome. Fingerprinting, targeting, profiling, or is it processing? Which one? Actually, direct profiling. Oh, the answer is processing. Right, most of the people actually answer fingerprinting, but the answer is processing. All right, let's see the leaderboard now. Is there any change? Wow, Angel Ganur is coming up out of nowhere. Okay, let's see the next question. When do we use targeted methods in metabolomics? Is it when we have enough time, or when we do not have enough prior knowledge on the pathway associated? When we have good support for chemical identification, or when we understand metabolic enzymes kinetics? Let's see the answer in. There you go. The answer is B. When we understand the enzyme metabolomics, the metabolic enzymes and the kinetics. Let's see, is there any change to the leaderboard? And Dian is leading with almost 50 point difference from the second place, Li Tanhui. I'm still wondering which Dian is this. Okay, let's go to the next slide. So the answer is which are following metabolic research flow except sample preparation, data processing, validating, or acquisition? Which one? I think we can all agree which one is definitely not the answer. But which one is the answer? Okay, data validation. We do not do data validation. We do data normalization, but not validation. Okay. Wow, Angel Gano is actually surpassing Li Tanhui to the second place, but Dian is still in the first place with 431 points. Okay, let's go to the next question. These following tools or instruments are widely used in metabolic research, except is it infrared, mass spectroscopy, X-ray spectroscopy, or nuclear magnetic resonance. So IR, MS, XRD, or NMR, which one? Okay, that's okay. Sigel will send the quiz link to the Telegram. Alright, the answer is XRD, X-ray spectroscopy. Okay. Most people get it right. And still there is no significant change to the leaderboard. Yes, we will share the quiz later on, Sigel. Don't worry about it. Okay, the first place is still Dian, the second place is still Angel Gunner, and the third place is still Li Tanhui. Now, the last question. Which one is not an application in the industry? Self-improvement, quality control, contaminant, or halal food? If you have idea what halal food means, it's basically kosher for Muslim. I'm sure most of you know what halal food is. The answer is A, growth improvement. Okay, let's see. What is the final score? The final score is there isn't that much change, but Dian is still in the first place with 584 points. Angel Gano in the second place, Diandra in the third place, Li Tanhui in the fourth place, Tahlah in the fifth place. Okay. Can I ask which Dian is this? We have two Dian in the Zoom meeting. So we have Dian Mukdalifah and Dian Nugrahaini. Can you please raise your hand? Which one is you? Which one you are?
Maybe Dian Nugrahaini.
Dian Nugrahaini?
Yes. Yes.
All right. Congratulations on getting the most answer correct for this session. I hope you can do it again in the next session. Okay.
Okay. Thank you. See.
Okay. Thank you, Dian. Okay. Okay. So after this, we still have until 12 o'clock. So first, I would like to remind everyone, we still have peer assessment in Gather Town. So if you have not yet give a peer assessment for today, please give a peer assessment. I think you still remember the link. I'll just share it right now. Okay, here it is. So if you have not yet, here's the link for today. And next, Bu video. What is the next game? I forgot about it. I'm sorry.
Actually, if the participants wants to share their thoughts or their comments or their suggestions, right. Otherwise we will share the link for attendance and peer assessment for today.
Right. Okay. Is there anyone who want to share anything about perhaps about their SFP or any other question regarding this program? Please raise your hand and open your mic. Anyone? I'm sorry that my camera is a bit grainy because I'm in room and my light is not that good. So sorry.
Oh yeah, Pak Hafiz, I think there is also another information. I think from the committee has already
Oh yes,
sent,
please.
yeah, I think the committee has already sent the email to to those who have best posters. I think if I'm not mistaken, it should be around 10 best posters. So those who received the email, or maybe if you haven't received the email for the best poster, so yeah, please wait. We have chosen the best 10, and for those that we will be required to present on Wednesday. Yeah, I think the committee hasn't sent the email, but there will be around 10 presenters who will present on Wednesday. So we will choose the best, those who have the best poster. Yeah, just to prepare. So I think everyone should prepare for this. And I do believe everyone has met their best for making the poster. So we really appreciate them.
Okay, I think that's all, Bu Amel. Perhaps you have anything else to add?
Okay. I'm sorry for the interruption because I'm in the outside. Okay. I want to complete the information from the Bu Fidi. Today, I will send the email for the best poster. Maybe if you want to share also, but you didn't choose for the best poster, you can, sorry, you can register for us to present your SFP, maybe to share about your research or your interest topic to us for the final session, yeah, in Wednesday, I think. So I will send the email within the night today, and you can reply my email for the SFP presentation. Yeah, maybe like that, Pak Hafiz.
Okay, thank you, Bu Amel, for your information. So let me reiterate the information that have just been delivered. So first, we have chosen the top 10 SFP, your which is part of your assessment, and those top 10 would be, we hope, would be presented on our last session on Wednesday, which is on the 14th session. And then if anyone want to also present their SFP, you can just contact any of the committee. So all of us are in the Telegram group. So if you have any question, if you want to volunteer to present your SFP, you can just please contact us. Okay, I think that's it for this session, Bu Vidya. I think we can share the attendance and the peer assessment link for today.
Yeah, sure.
Okay, so there's the attendance link. The attendance link is bit.ly VSS attendance 9. So talking the attendance link have been shared in the chat room if you are looking for it. So you can just click it. Okay, and also the link for gather town and for assessment is also present on the chat room and have been shared in the presentation over there. You can look at it. Okay. All right. For our next session, we will talk more about natural product chemistry, which is come from the sea. So far we have talking about natural product which came from legumes or beans. Basically, we have talked about natural product from Artocarpus, which include jackfruit, and some other also from jamu, and most of them is on the land. But for the next session, we are talking about natural product from the sea. So stay tuned, and we'll see you at 1 p.m. Thank you.
Yeah, so thank you everyone. Have a good lunch and please come back at around
Last night. Okay. Right. Welcome back, everybody, to our virtual summer school program. As I've said before, did my internet connections? Okay. As I have said before, today we are this time we are going to talk about marine and natural product from Indonesian water, and our speaker is Dr. Mastaria. He's not here yet, but hopefully he'll come soon. But first of all, I would like to talk to some of you who's already here. Let's see who's here. Putri Fatmawati, are you there? Hello, Putri? Okay, I don't think she's there. Let's see. What about Sekolah Tinggi Ilmu Kesehatan? Who's this? Who is Sekolah Tinggi Ilmu Kesehatan Nasional? Hello, Sekolah Tinggi Ilmu Kesehatan Nasional. Can you open your microphone and camera, please? All right. I see some familiar name here. Nur Afikah Manan, I think she was here last year. And then Edwin, of course. Pak Mastaria is already here. Selamat siang, Pak Mastaria.
Selamat siang.
All right, glad to see you here. Thank you for joining us. Thank you for agreeing to be one of our speakers in this virtual summer school program.
Thank you very much. You're welcome. So thank you, I said, for invitation for all the organizing committee.
Yeah. So last year we also did this program actually. So, but last year we talked about natural product and craft. So we don't really talk about all natural product. But this year we already we don't talk about craft. We just focusing on natural products. So thank you once again for your availability to be a speaker in our virtual summer school program. As I understand it, you work in marine natural product, right? So
yes, my research topic, marine natural product.
Yeah. Just a quick question. Are you also a diver?
Yes. I've licensed for scientific diver from Europe. Yeah.
Wow, that is actually very impressive. For me, at least, because to be honest, I cannot swim. So, so when I heard that one of our speaker is someone who is well versed in marine natural product and which is you, and he will talk about it, and he is a licensed diver, right? I'm just like, oh, I hope I'm not going to sink this time.
You don't need able swimming for do the diving. So yeah. You don't need swim.
Yeah, yeah.
If you want to get the diving license, you don't need to swim. But one of the, you just need like using the scuba snorkeling, like you just need 200-meter swim, something like that, using the gear of the scuba snorkeling. So if you want to do the recreational diving, so many people in Bali and Lombok doing like that. So
I
yeah.
see.
There's a professional instructor that can help you and accompany you during your go to diving.
Have you dive anywhere interesting?
Anywhere
Somewhere
interesting?
maybe Raja Ampat or Wakatobi, perhaps? Have you dived there?
I've been several places in Indonesia, from Sabang to Biak. I've been there, Raja Ampat also. What is the interesting place? Yeah.
Well, for me at least, anything anywhere the sea would be very interesting because I've never been there. I can't swim, just as I said before.
I think, yeah, like a Bunaken also interesting because they are wall, yeah, coral reef walls, not slope coral reef. I prefer to dive in the wall coral reef, not in the slope, because yeah. And do you know where's the place Spermonde in Makassar? That's also interesting, also for diving also. And
I see.
And where's the place? Yeah. Komodo also nice place, very interesting also for diving. But mostly, most of my favorite place maybe Bunaken, because first I got my lessons there. The first time I did divings there, and every years I went there. So, but not in the pandemic times here, so it's difficult also there.
Yeah.
And where's the nice place? Yeah, maybe Sangihe in North Sulawesi also, because there is a volcanic mountain inside the sea, so you can find with the sea with the hot temperature there. So because under the sea there is the volcanic mountain, and yeah, that's also very interesting. One the place is also in Sabang, in Aceh. There is there also the
really?
Yeah, there's volcanic mountain there, volcanic mountain in under the sea. So that's very interesting. So, but mostly when you interesting with the biodiversity is Indonesia is very abundant for the marine organism, more than Indonesians, West Indonesians, because difference deep, yeah.
Nice.
So in East Indonesia is very deep, but in West Indonesia not really deep, so not interesting for the marine organism for living there. Even in Belitung, so Belitung is not interesting also to do the diving. So you just find a coral reef, not a sponge and soft coral, something like that. So but on the beach, Belitung is very beautiful. But to go diving there is not. I think it's not my favorite place to go diving there.
What about abroad? Have you dived abroad?
I've been dive in Florida, and also I've been diving in Mediterranean also. Because my PhD in Italy, so sometime I went there to Mediterranean and to do diving. Then my postdoc in Florida, so I've been in Florida Keys for do the diving with my former supervisor there. So yeah, but my favorite place always Indonesia, because we are very great diversity for marine organisms.
Yeah, we have like we are the biggest archipelago nation in the world, so I do expect that we have like the most diverse marine biodiversity. Is that correct?
If we with the terrestrial and marine organisms, yeah, if we combine both of them, I think we are the most we are the greatest we are the greatest biodiversity in the world. But in terms of the terrestrial, still Amazon, Brazil is number one. But in terms of the marine organisms,
Yeah.
still Indonesia is number one. But if if we combine both of them, so I think we are the greatest biodiversity in the world, I think.
I see. You see. Uh, uh, when I'm thinking about biodiversity, most of the time I'm actually thinking about fish, not really about plants. So even when I'm thinking about plants, most of the time I'm actually thinking about algae or coral, something like that. I'm not sure I'm thinking about the right biodiversity. But there is also sponge, right?
Yep.
Sponge. Also, wait, is sponge? No, I don't think sponge is. Yeah, sponge is porifera, so it should be animals, not plants, right?
Mostly animals. In the coral reef, mostly animals.
Mostly animals, right?
Mostly
like,
animals.
just like
The plants only, yeah.
anemone.
Yeah, mostly
Anemone is
animals.
also
Mostly
animals, right?
invertebrates.
Yeah. Okay. I think we should just start this session. We have like 3 minutes left, and the participant is what, 20 something. Okay, I think we can just start this noon session. All right, once again, good day, everybody. Welcome back to our virtual summer school program. My name is Hafiz Haji Aziz. I'm your MC for this session, for today's session. So in the first session, we talk about metabolomics, so of natural products. And then for this noon session, we have Dr. Mastaria over here, and we will talk about marine natural products. So thank you, Dr. Mastaria, for agreeing to be one of our speakers. Bu Vidya, can you please display the CV?
I think we will have a video first.
Oh yeah. All right. Right. I forgot. First, before that, we would like to show you a video on marine natural products. Most of us, not most, but all of us is an amateur in marine natural products. So hopefully this video would be a satisfactory introduction, glimpse into marine natural products. So please enjoy. All right. That is a short glimpse of marine natural products from the sea that we have gathered. I hope we didn't make a big mistake over there. But even if we did, we have some an expert here that would straighten things up for us. So let me introduce you to our speaker for this second session for today. So his name is Dr. Mastaria. He is a marine biologist, specialized in marine natural products, drug discovery, medicinal chemistry, and chemical biology. So his bachelor degree is in Universitas Andalas. His master and doctoral degree is in Mars Polytechnic University in Italy. That's why he had dive in Mediterranean. Next. Right. This is just some of his research. So most of his research is about marine products, as we expected. So we have microalgae derived like sugar, and then screening of antioxidant and anti-acne activities in sea cucumbers. And then there is also antioxidant and antibacterial activities in sea cucumbers. And the last one is anti-infection and antiviral activity, also from sea cucumbers. He likes sea cucumbers, it seems, because it's interesting, of course. He also have some interesting achievement. The 8th Travel Awards edition in Global Young Scientist Summit in Singapore, postdoctoral fellowship from Fulbright programs, also Best Oral Presentation Award in International Seminar on Indonesian Society of Microbiology, as well as Travel Award from Società Chimica Italiana, International Summer School on Natural Products. So it seems like this is not his first summer school. So without further ado, please, Dr. Mastaria.
Okay, thank you very much, Pak Hafiz. Good afternoon, everybody. This is first of all, I would like to thanks to all the organizing committee for their invitations, for joins as a lecturer for the International Summer Schools of Natural Product. So that's very interesting for me. Because this is the first time I make a lecture for summer course in Indonesia. So I'm a little bit curious because first times I'm not like not a famous natural product chemistry in Indonesia, I think, but people's in the University Pendidikan Indonesia, in University of Education of Indonesia, invite me for the lecture about marine natural products. So I try as best as I can do in terms of my experience in natural product chemistry. So first of all, I will like to introduce for you myself. So my name is Masteria Universa Putra. So my PhD degree about the marine biotechnology. So in 2000, I got my PhD in 2012 in the marine biotechnology, especially searching the bioactive compound from the marine invertebrates, sponge, and soft coral. So I'm also a chemist because I got a graduate, my bachelor degree in chemistry So I combine my expertise and my experience between chemistry, biologists, and ecologists. So to combine with the research of marine natural products. So let me share my presentation. So as I mentioned, Pak Hafiz, today I will give you some perspective about the Indonesian marine organisms as source of natural products. Mostly people's Indonesia, I think in Southeast Asian, still working with the terrestrial natural products. Not many people also in Indonesia working with marine natural product. I think the pioneer in Indonesia is from LIPI and also from the University of Diponegoro and also from the University of Gajah Mada, I think. So the first people working in marine natural products in Indonesia. But most people working in marine natural product Indonesia is foreign researchers, and some of them collaboration with Indonesian researchers. But now since 2019 and 2018, most people in Indonesia interesting to search new bioactive molecule from marine organism. So I give my lecture in the fourth chapter or for outline. The first about the general introductions about the natural products, why sea or marine environment produce the marine produce natural products, and the pharmacological activities interesting from natural products. Then the second part will explain to you with some of our research in our group, the result of our research in our group about the soft coral and tunicates. Then the third part about the sponge. I think this is famous object for natural product in the world. I think in the past 5 years, but in the past, but in Science 2011 and Science 2012, marine microorganism is very interesting people to study natural product. And the third part for the bioactive natural product from sea cucumbers that Hafiz mentioned, because I'm personally interested with sea cucumbers right now. I working in the 3 years ago about the sea cucumber. Then we collected mostly, I think we are the research group that have the more collection of sea cucumber species in Indonesia. We have 51 species collection of sea cucumber from the Indonesian sea cucumber. So let me introduce first. So as you know, that's the ocean. So we call it here Earth, but mostly in the Earth, more than 70%.
Kita nunggu Pak Bambang, ya. Silakan boleh.
Okay. More than 70% of the Earth's surface is the ocean. So this is interesting because only 30% in the Earth's mainland. So and 70% on is the oceans. And the ocean contains more than 2.2 million marine organisms. So I think if we compare with terrestrial, I think the marine organism most abundant from the terrestrial or from the terrestrial. And mostly we can, coral reef is the mostly home for 20% of marine organisms in the ocean. So that's why, because what? So as you can see that this is the deep of the ocean. So mostly the deepest point in the ocean is Mariana Trench, more than 11,000 meters. But the coral reef here, around 40 meters. So 0 meter for 40 meters. So that's why, because they can able for the photosynthesis and the suns to come to 40 meters, and the humans can survive by scuba diving with 40 meters. That's, we call it the coral reef. And yeah, this is where we can see the deep, like the deepest blue whales dive. So we can see the blue whales of 500 meters. So if you compare with the Grand Canyon, more than 8,800 meters, something like that. But if we talk right now about the marine natura, mostly of the people research about the marine organism in 40 meters. But in 10 years ago until now, mostly people interesting also with the deepest of marine organisms. So, and we did some of the expedition from in several area in Indonesia to collect the marine organism in the deepest Indonesia, based on the East Indonesia, and we collected marine microorganism and marine organisms there. So, but mostly our research in our research group on coral reef. So, as we see in the perspective of the biologists and ecologists, that's why the coral reef is very interesting. So, if you're able doing snorkeling, or if you're able doing diving, you can see in the coral reef there is several organisms. So such as soft corals, so the algae, some microalgae. So sometimes microalgae have the symbiotic with the hard coral, sponge. So that's the sponge. So every day we watch SpongeBob, but here we watch in the coral reef, it's the really sponge. And tunicates. So a tunicate, we call it the ascidians also, and the mollusca, and gorgonian, a fish, and diatom fish. Mostly the coral reef that we eat every day here is coral reef, fish from the coral reef. So that's it. So that's why the interesting of the coral reef to study of the marine natural product. So as the perspective of the chemist, I'm the chemist. So even my graduate of marine biotechnology, so I studied also the ecology, but I'm still a chemist, and my postdoctoral medicinal chemistry, and starting doing the biopharmacies also. So, but my perspective, my point of view about the natural products, about the marine organism, so marine natural product, why the marine organism produce the organic compounds. So that is my perspective point of view. When I look the coral reef, I look the organic compounds. Though I'm not looking the sponge, I'm not looking the ascidian, I'm not looking the soft coral. But when I see the coral reef, that is the the place where's the organic compounds abundance here. So I see here. So now in the markets, there is 8 marine natural product already approved for such of treatments like anti-cancer, so also antiviral, also anti-inflammatory, also the cardiovascular also, and mostly from the FDA approved, there is the anti-cancer compounds from the marine natural product. So that's I will explain to you why mostly the marine natural products in the market for the anti-cancer. As I mentioned before, that's now in the past 5 years, people interested with marine microorganism, not in marine organism right now. Because what? In the coral reefs, such as sponge, soft coral, and tunicate, they are filter feeders. So they eat with the marine microorganism. They eat the bacteria for the growing. So that's why people interesting how the marine microorganism can survive in the coral reef. So here mostly we can combine with invertebrates like sponge, soft coral, tunicate inside these invertebrates. There is only in 2017 or in more than such as more 500 novel compounds. But if we combine, if we talk about marine microorganism, almost 900 organic compound from the marine microorganism. That's isolated also from sponge. That is also isolated from the sediments, tunicates, or soft coral, or what else, or mangroves, something like that. So that's very interesting. So this is very fantastic for the marine natural product chemist. Right now we have the phase 3 clinical trial for the anti-SARS-CoV-2. So as mentioned before, this is plitidepsins. So plitidepsin have the approved for the anti-cancer from the European Agency for Drugs. But some of the company called it the PharmaMart, they make the repurposing drugs from the anti-cancer for the anti-SARS-CoV-2 for the COVID-19. And now they are entered the clinical trial phase 3 for the COVID-19. And these compounds more potent than remdesivir, also more potent than the chloroquine. So hopefully in the next year or this year, they will be get the approval for the clinical trial plitidepsin using for anti-SARS-CoV-2. So I mentioned to you before that anti-cancer have the great interesting for the biological activities or pharmacological activities for the natural product isolated from the marine organism or microorganism. So see here, we compare with the cancer and other disease, mostly compound that isolated from marine organism have been examined for the anti-cancer activity. And this is the U.S. National Cancer Institute also mentioned that 1.8% of the extract from marine animal have shown anti-cancer activity compared with terrestrial plants. So that is very interesting to study the anti-cancer compounds. Mostly people interested with marine organism or microorganism right now than the terrestrial. So that's why the marine organism of microorganism produce the anti-cancer compounds, because what in the coral reef there is a competition of space, because there's like a sponge, soft coral or ascidian, they cannot moving, so they can't talking each other with the language of the organic compounds, with the language of chemistry, not with they cannot speak each other like human. They can speak each other with the language of the chemistry. So because they are kind of moving everywhere, so they are static. So there is the difference. They have prevents the place from the competition in space. So if they prevent the place for the space to the others animal, to other organism, they produce the chemical compounds that have the cytotoxic to make the other competitor died or to make the other competitor lose. That's why mostly the marine organisms produce the cytotoxic compounds. So first, for defense from the predator, so because they cannot moving everywhere, so they just stay there, and then prevents for the competition of space, and also find a match to extract it. So and then for protect themselves from infection. Why for protect themselves from infection? Because mostly the sponge, like from ascidian, tunicate, and organism in coral reef, they filter feedings and they eat to growing to eat the microorganism. That's why they produce the cytotoxic compounds. And if we talk about what is more promising between the marine organisms or invertebrates than marine microorganisms. So this is what one of our colleagues, De La Calle from the PharmaMart, that mentioned that if we talk the marine organisms, wide range of chemical defense. So they produce so many organic compounds with toxicity, but it's the industrial supply is very challenged, because what if growing the sponge or for tunicates, that takes so many years for growing. But soft coral, we can doing the marine culture and they can growing only 3 months or 6 months. But for in case of sponge, they will growing for many years. And the other options about the microorganisms. Microorganisms, that's very fantastic. We can do the fermentation with large culture fermentation. But one of the challenge is more than 99% non-cultivable. That's the challenge for working that. And also the other option, metagenomes or biosynthesis natural products. But the problem is to solving to get the heterologous expression is very difficult. Takes time, takes challenge, and takes emotional, because most people working with the biosynthesis, they can produce paper 3 years or 4 years. That's people working. That's not much people working in biosynthesis natural product right now. The other challenge that they make doing the semi-synthesize. So if you're talking about the semi-synthesize in pharmaceutical developments, there is to first synthesize. That's one of the other options. Then semi-synthesize, and now biosynthesize. But mostly the pharmaceutical company interesting with semi-synthesize because this low cost than the synthesize, because synthesize you need less than 50 steps to get the in the market to feasible in the market for your synthesis compound that we are producing, and very costly. And biosynthesis takes time to get the DNA to produce the compounds. And the last option is semi-synthesized. So we combine with the production such as microorganisms. Then we make the semi-synthesized there with the other synthesized with the step less than 20. And that's in the market is the drugs is very cheap. So that's why in the market right now, most people challenge to get the compound, and the expensive, the anti-cancer compound, or mostly drugs in the market is very costly because synthesized is very expensive. And that's why the combined with the semi-synthesized to the viable in the markets. And what is discovery platform for marine natural products? That's my dreaming. So what in my head, how can in Indonesia also can produce one compound or one lead compound to be in the market as anti-cancer maybe, or as the anti-inflammatory maybe, or what else, everything biological activities. So the first step, what we need to do is the expedition. That's really important. So because Indonesia is the archipelagic country, we have 80,000 islands in Indonesia. So we are the most greatest marine biodiversity in the world. We need the collection. We need the expedition. That's very interesting. So we need to collect more the marine invertebrates. But don't forget with microorganism. Microorganism is very important. So we combine with marine invertebrate and microorganism. So the other things, that is cell biology for anti-tumoral screening, because I'm interested right now for anti-cancer. That's why one example for me is for the anti-cancer. So you need to do people with the cell culture, in vitro culture, sample preparation, cell seeding, cell treatment, quantification, and evaluation. That's very interesting. That's very important, because this is the first time for you the screening of your extract, or the screening of refraction of the screening of your compound. That's the first step, the critical step for you to develop of drugs. Then, if they're an active mechanism of action, preparation of compounds and region for analysis, molecular biology assay, cellular assay, biochemical assay. And the next step is synthesis process. It could be semi-synthesized. You find analog from the from bacteria, or what else? You find analog from the market that already produced the compound, and you're doing the semi-synthesize to get the lowest step for the synthesize. That's very interesting. And if they're the viable in the market, or the process step of synthesis not very expensive, you can develop the pharmaceutical developments. And the most important is in vivo and human trial. That's very important for you to discovery platform for marine natural products. This is what we call the first. So you need the collections. So classification, storage, taxonomy, barcodings, and field data analysis, laboratory preparation. That is very important. The first important, because why the collection is very important? If you collect in sponge in one area, it could be difference of the organic compound, could be different secondary metabolite they produce, because they are make adaptation with the environments. So that's a very important that we need the collection, and we need the field data for the marine, from the marine organism that we collected. Microbiology, so that's the very thing. If you collected the marine invertebrates, don't forget to collect the marine biology. So if you are microbiologist, I think most people here, I think you study, make a practicum, laboratory practicum with microbiology. So you do the isolation plate, get a pure colony, doing taxonomy with 60 sRNA. If there are new compound, you're doing with the whole genome sequencing, something like that. So that's very important. And extraction. So extraction is traditional method. So you can do with the organic solvents, with methanol, dichloromethan, or ethyl acetate, most of them. So, but for me, if I talk with marine natural product, I prefer doing combined with methanol and dichloromethans. So polar and non-polar compounds. Then doing the bioassay. What is interesting for the bioassay? If you are interested with the anti-cancer, so use the human cancer cell line, such as colon cancer, lung cancer, human breast cancer, or pancreatic cancer, or what else you're interested? If you are interested with anti-diabetic, or you're interested with anti-inflammatory, you can use the inose protein inhibition, or like COX-2 or TNF-kappa B, something like that. So do what kind of interesting for you the biological activity. After you get the extra or the interesting fraction that have the biological activity, do the isolation or structural elucidation of the bioactive molecule. So one thing that you interesting here, I think you already talking about the metabolomics in the morning, right? With Parafi, right? So here, that's also the interesting about the metabolomic, or we call it the replication. So the replication here, you can use the NMR, you can use mass spectrometry, and you can search based on the mass spectrum molecular weight or your NMR in database like MarineLead, SeaFinder, and GNPS. So you're not wasting your time to isolate that non-molecule, or if you're interested in the new molecule, you can get from the information, the replication in database that you can, oh, this organism produce new molecules, so I can isolate make the isolation for the new molecule and make the structural elucidation using NMR or using the LCMS GitHub, something like that. Yeah, that's very interesting. So you need to understand spectroscopy. That's the very important and new for structural elucidation of the organic compounds. And the others, that's one thing that people asking me, there is marine natural product database. Why people working? Yes, there is marine natural product database. This is MarineLead. MarineLead is the marine natural database that developed from Professor Blance from the University of Canterbury in New Zealand, and now they are cooperation with the Royal Society of Chemistry in England to get the marine natural database. In marine natural database, it's very easy. From the molecular weight that you get it from the LCMS, just put your molecular weight and put your name of your species. You can know that's from the molecular weight that you get your compounds known or new. Even the new, maybe even they have in database, we can check also there is the different stereochemistry right now or not in NMR. So that's very interesting. That's very easy for you right now if you're working with marine natural products. So then there is the global platform natural product chemistry for LCMS-MS. I think not much people in Indonesia use the GNPS because the GNPS first they built for marine natural product from the Professor Peter Dorestein in University of California, San Diego. But now people put all of the data for LCMS-MS from terrestrial also. So if you study metabolomic or if you want to study metabolomic, you should understand Global Natural Product Society molecular networking. because most people right now in the world put the LCMS data in this database. So you can find, or you can understand the three organic compounds that you need, that in your extract or the fraction. You can know that there is new or none also. So I think if you are the organic chemistry or natural product chemistry, that's the very powerful database that you need to learn. So that's database. And yeah, as I mentioned before, if we looking like, if first the Discover platform that I mentioned before, if you already got the data for in vitro, in vivo, and you have the isolation from macroorganism, but there is have the not expensive for the market, for the synthesize. And if you have the compound confirm the in vitro in vivo for fermentation, so you can continue your compound to be the drugs. So investigation and new drugs. So what else? The feasibility of biochemical synthesis, that's very important. Negative compare the analysis and different mechanism of action. Why the different mechanism of action very interesting right now? For example, for the anti-cancer compounds or drugs in the market with the anti-cancer right now, FDA doesn't want to approve the drugs with the same mechanism of action for the anti-cancer. They want different mechanism of actions. So test people right now, if they're active for the cancer, they still don't know that can be approved for the FDA. FDA want to do different mechanism of action. That's why people interested with medicinal chemistry right now, to find the new mechanism of action, not to find only the new anti-cancer, but to find the new the mechanism of action. That is very interesting. And let's talk about Indonesia. So that's I mentioned it before, that Indonesia is very one of the greatest marine biodiversity in the world. So we have the archipelagic country. So I think we have more than 80,000 islands right now. The three big islands in the world in Indonesia, like Java, like Sumatra, and Kalimantan or Borneo or Papua. There's three the big islands in mainland, the big islands in the world. So in terms of the marine biodiversity, I think we are the #1 in the world right now. So we have the sponge, more than active, I think 850. So we have marine fish, more than 3,000. So we have the algae around 700 species. And we are the complete seagrass in the world. So we have all of the seagrass in the world we have in Indonesia. Then we have the mangrove. So in the mangrove, we are very I think we are also in the complete the mangrove plants in Indonesia compared with the Middle East. Yeah, in Middle East, I think there is 1 or 2 mangrove plants growing in the Middle East, but Indonesia we have 43 mangrove plants. So marine natural product Indonesia, I think the first publication about the marine natural product Indonesia in 1988 from the Kearney, from the people from the University of California. So they isolated this compound named Hiatella, so laulilamide. So this compound named laulilamide. So this compound have the cytotoxic for the anti-cancer, and most people working with this compound, so antimicrotubule. So I think there's still this compound have in phase 1 clinical trial right now. So but the first compound, the first organism that isolated this compound from Indonesia. And until now we have more than 732 marine natural product, novel compounds of marine natural products that isolated from Indonesia, mostly from sponge, tunicat, and soft coral. And that's very interesting. So most of right now, several marine natural product in Indonesia shown the biological activity or pharmaceutical activities such as anti-cancer, anti-inflammatory, antibacterial, antiviral, and so on. And what interesting is we sometimes we make a name based on where we collected the species of the marine organism. For example, you notice manu peroxide, so they collected from Manado, and this is have the activity from antiplasmodium. So this is Bitungolide that I've collected from the Bitung, also from North Sulawesi, and Lembehine that collected also from Lembeh in Bitung, also in Lembeh, in the Lembeh Strait, and some of them Biaketidae isolated from Biak, Papua, and Papua Namidae. And Papua Namidae have potential for the cytotoxic compound for the anti-cancer. And Teriswin holiday A and Barangamidae A that's isolated from I think the name of the place is Baranga or Barang. So this is the place, Pulau Barang, I think, then the name of the place to collect it. And that's very interesting from the Indonesian marine natural products. And our research group recently published one paper about one review paper in the molecules. So about the marine invertebrate from Indonesians from Indonesian marine natural product from Indonesian marine invertebrate. So from our papers, you see here mostly natural products produced from the sponge. So sponge still the number one organism produced natural product in Indonesia, and the second one is soft coral and tunicates. But you see in the map that we make here, mostly I think most 99% natural products came from the marine organism that collected from East Indonesia. We still only have one compound or two compound from the East Indonesia, but this in Jakarta. Why Jakarta is very interesting? Because we are more pollutant and produce more molecule than the other place in West Indonesia. And for the activities, mostly the cytotoxic activity from sponge, soft coral, or tunicates, something like that. So here, and what we did in my group. So right now, that's I mentioned before that my group's interesting with marine natural products. So we development a liberate natural product from Indonesian marine organisms. So we collected marine microorganisms for several places in Indonesia. So we collected from Sabang also, but we only have the collection from until Biak. So we still don't have Merauke. Hopefully we can collect it in Merauke in the other time. But we collected also marine microorganism. So marine microorganism, we start doing that in the past 3 years ago or 2 years ago. We have some publication here, but I am not sure in this presentation, but we have the 3 paper about marine microorganisms. So that's what we're interested in the lab. So we want to be the center of the very natural products for marine organism in Indonesia. So this is one of the first compound that isolated from marine organisms. So soft coral sinularia. So the first marine organism that I did to isolate the new compounds. So here from storidal fraction from the sinularia, you see here we got the 5 new compounds and 3 non-compound. So one the new compound is gorgosterol, one the non-compound is gorgosterol. So gorgosterol is the major compound from all of the soft coral, even the sinularia, even that sarcophyton, even that lobophytum, everything. That if you isolated the compound from the soft coral, I believe 80% you can isolate gorgosterol. But in our case, we collected also 5 new compound from the sinularia, and one new compound have the cyclopropane ring, the same with gorgosterol. And this gorgosterol have the potential for the treatment of cholestasis. So we analyze that with the farnesoid X-activated receptor, and this gorgosterol have shown the potential. So cholestasis treatment is very important if you have the disease of liver. So that's very important, and we still continue to study with gorgosterol, with the semi-synthesized, something like that, that we can produce more activities, this gorgosterol, for the farnesoid activated receptor. And the others, so we isolated several compounds also from the sinularia. So here we isolate from the non-polar compound, we isolated five compound. Three of them the new compounds. We call it the chloroscabrolide A because they have the CCl chlorine ring here, and two non-compound with scabrolide D and leptocladolide B. And this chlorine ring, I think the second compound from the sembranoid that reported in the literature right now. And the other, from the polar fraction, we isolated two compound, one of them new compound, and this is very interesting. Why? This is very rare in nature because there's the acetylated acetyl ring here attached with the glycoside. That's very interesting because most of them from semi-synthesis like acetylation synthesized, but in our case we isolated 3 ring, and that's very rare in nature. And from the another fraction of the from polar fraction, we isolated 2 new compound named sinulalfoxide and sinulalsulfone. And this is the alkaloid compound, but they have here the sulfur ring here, and that's very rare also in natural in nature. And most of them we evaluated with anti-inflammatory using ionized protein inhibition. And we can see here sinulalfoxide shown the moderate activity, and sinularicide and seborrheicidase also show the moderate activity for ionized protein inhibition. And the other compound, 2 compounds that we isolated here, from the soft coral sarcophyton. Sarcophytoxide and sarcophin is the major compound from the soft coral sarcophyton. So if you're doing the extraction and you using the column chromatography with first with the NXan, you can directly isolate this compound. Sarcophytoxide D and sarcophin is very polar compounds. And we tested here with TRPF3, and TRPF3 is the neuroactive or anti-inflammatory activity for the receptor. So we isolated it and we evaluated with TRPF3, and here we saw that most sarcophytoxide and sarcopin have the strong activity for the TRPF3. And this is very interesting. Licheta is one of the first compound from sponge that I worked in 5 years ago, 6 years ago. So Licheta is most abundant in Indonesia, the small or tiny, very tiny sponge. So and the major secondary metabolite that produced from Licheta is lichetamol A and lichetamol B. This is number 1 lichetamol A, and number 3 is lichetamol B. And 2 papers mentioned that lichetamol I and B have the potential for antimicrobacterium or anti-tuberculosis, and the other papers showed that also the lichetamol have potential for the ubiquitin receptor. In our case, lichetamol that we evaluated this with TRPA1 and TRPM8 channel. So TRPA1 and TRPM8 channel is very interesting for neuroactive also, and also for the inflammation of pains. That's very interesting for the using for TRPA1, TRPM8 channel. And we cannot publish with the non-compounds, but we need to publish with the new biological activities. But we studied also with structure activity relationship of this lichtamol A and lichtamol B to understand this mechanism of action of the lichtamol A to as modulator for TRPA1 and TRPM8 channel. So we did a study for structure reactivity. So we start semisynthetic, so simple semisynthesis. For example, we did the acetylation semisynthesis, and then we do the reductions semisynthesis. So that's very simple semisynthesis. So for example, for the acetylation, we change the wedge ring here to be the acetyl ring here, and for the reduction, so we change the double bond here for the single bond here. So that's in the simple semisynthesis that we did here. And for the semi-simple semisynthesis here, we can produce 6 semisynthesis compound, and then we evaluated for TRPA1 and TRPM8. So as we understand here in the semisynthesis here, you can see this is inhibition concentration for TRPA1 and inhibition concentration TRPM8. So if we change the OH group here to be the acetyl group, the decreasing the inhibition concentration. So here, the increasing the inhibition concentration. But if we change the double bond to be the single bond, then not much more increasing for the inhibition concentration. That means that the OH group is very important for the mechanism of action TRPA1 and TRPM8. So that's why I think that's why the biological activities of the lichtamol A and lichtamol B have potential for neuroactive and anti-inflammations. And here that I mentioned. And the other works here, that's tourniquet. So most of you tourniquet. So if you are being snorkeling, if you are able to snorkeling or diving, snorkeling, I think in everywhere in the world except Arctic, except Arctic, you can find the polycarpa. So the polycarpa is the major, I think the major ascidian or tourniquet in the world. You can find it everywhere in the world except the Arctic. So the polycarpa, and you can find also if you go to only swimming in the coral reef, you can find also the polycarpa in 1 meter or 2 meter. And this polycarpa, we isolated two new compounds named polyaurine A and polyaurine B, and we evaluated for Schistosoma mansoni for antiparasite, and this polyaurine have the impaired X production for the Schistosoma mansoni. And this is our work for the ascidian or tourniquets. And we have the collaboration with the pharmaceutical company in the world named PharmaMars, the famous pharmaceutical company that they are only working only for the marine natural product. And interesting with anti-cancer. We did the first expedition in 1,000 islands in 2 years ago in the 1,000 islands. So we collected more than 100 active 19 sample from the Bryozoa, Cyanobacteria, Cnidaria, Echinodermata, Sponge, Mollusca, Polychaeta, and tourniquets. And we evaluated all of them with anti-cancer, MDA, pancreatic cancers. We evaluated also with lung cancer and the colon cancer. And from 119, only 245 have the potential for the anti-cancer compound, have the activity for cytotoxic. But when we do the replications, only 50 sample have potential to get and to have the new compounds. So that's what we are following for all of the compound, all of the organisms that we collected. So we only doing the extract only 2 gram or more or less, and doing the traditional extraction, and we screening with the MDA, F549, HC29, PSN1 or pancreatic adenocarcinoma cancer. And then we doing the replication to get to find them of new compound or non-compound inside our extract. So we know that most of them have the non-compound, but we interesting the new compound with the new anti-cancer activity. That's very interesting. That's what our main goal for doing the extraction. And we did a fractionation for all of them, only 41 samples that we discarded 41 sample, but we continue. I think we just right now we just continue from 119 sample. We continue only 5 samples that have the anti-cancer compound with the new compounds. So only 5 from 800, only 5 samples that could be continued. And from our researches, our first our publication last year in Journal of Marine Drugs. So from the sponge of Seribu Islands, we call it Indonesia, or 1,000 Island in English. So we selected 2 new compound peptide type of compounds. We call it seribunamide and haloericiniamide A. But unfortunately, after we got the new compound and we tested again this new compound with human cell cancer line, and there is no activity, and that's is very fantastic, and we still don't believe it, because from the extract and fraction shown the activity. But yeah, maybe the major compounds show the activity for the anti-cancer. And this is very interesting. This our publication this year already accepted. So we selected also, sorry, this is not soft coral but sponge. So we selected from the sponge 2 new compound type aditopronoid, but this compound show moderate activity with human cancer cell line. And we submitted this publication in Journal of Natural Product, and hopefully will be published soon or accepted soon. And this is what we are doing for sponge. And another interesting that I mentioned before in the beginning of my presentation, we interesting with sea cucumber. Indonesia is most abundant of sea cucumber, and also Philippines also most abundant for sea cucumber, and more people in I think in China, Japan, or Korea eat the sea cucumber for functional food. And I'm interesting to share the anti-cancer compound for sea cucumber, also for the functional food also, because what the sea cucumber have contained the major compound from sea cucumber, there is saponin compound or glycoside triterpenoid compounds, and most of them have the activity for anti-cancer. We collected until now 31 species sea cucumber in Indonesia, but we have finished to evaluate, I think, around 31 or 21, 33 or 31 species sea cucumber in Indonesia. We doing the cytotoxic compound using the sulforodamine method. In our lab we not use MTT assay because people come on using MTT assay, but in our lab we use the sulforodamine B. I think because more cheaper than MTT, that's why we use the sulforodamine B. And from the results, as I mentioned before, the sea cucumber very active for anti-cancer. So, and have the cytotoxic compound called the saponin. And I see here that only 2 compounds not the activity for the anti-cancer, and mostly the like a Boachia, Holoturia, and Stichopus that people in around China and Korea and in Japan that eat it, they have the activity for anti-cancer. And the other that we collected in the other in the other different area, so most of them also have the anti-cancer. And other area that we collected, because we collected in 3 areas in Indonesia, then we interesting why this sea cucumber have the potential, the activity for cytotoxic, because in our opinion, first, if we collected different in the different area from West Indonesia, East Indonesia, and center of Indonesia, maybe they can have different activity, but and even in the different in different environments, they always produce the cytotoxic compound, and that's very interesting. And to understand the metabolomic, or to understand the organic compounds that contain in the cucumber, we did the replication also. We did also the metabolomic. So we're using the LCA, HPLC, TOF. So, and we use the database. We also develop our own database, but we check it also MarineLead, also Masanta, and other commercial database also. And you can see here, that is very interesting. Why? And this is interesting also for the chemotaxonomy, not in classical taxonomy or the molecular taxonomy, but also interesting for the classical taxonomy, for chemotaxonomy. Because here, if you can see here in the species Holoturia, most of the species Holoturia produce holoturin A or holoturin B, and that's very interesting. And we see here, so that's very interesting. This is Stichopus hermanni. That's for us that we believe that this Stichopus hermanni, and based on the classical taxonomy, but in the chemotaxonomy produce the holoturin B. We don't believe it with the chemotaxonomy and the classical taxonomy. We did the molecular taxonomy, and the molecular taxonomy, this Stichopus hermanni, not the species Stichopus hermanni, but the species Holoturia. So that is very interesting for us. If we doing the chemotaxonomy, the species or genera Holoturia, they always produce the holoturin B. But in the species in genera, genus Stichopus, they always produce the organic compound named Stichchlorisidae A or Stichchlorisidae B. And that's very interesting. If you see here in our the replication, mostly I think 99% of the Holoturia have produced the holoturin A or holoturin B, and Stichopus always produce this Chlorisidae A, and this is very interesting. And this is the tetic compound from the sea cucumber, holoturin A, holoturin B. So that's very interesting because they have the glycoside ring and have the like tritopanoid ring here, that maybe that they produce the cytotoxic activity. But we already study why the compounds, this compound have the cytotoxic activity. We cut this ring only with the glucoside, only with tritopan ring, but together they don't have the activity. So that's mean the combined with the glucoside and tritopanoid ring here, that's the structure activity for the cytotoxic compound from the sea cucumber. Here's Stichchlorisidae A2 and Stichchlorisidae A1, as I mentioned before. And this is Holoturia. That's some of organisms that from our replication produce the organic compounds. And thank you very much. I think this is my final presentation. I think, yeah, one hour, right? My time. So thank you very much for attention. I deeply acknowledge for all of my collaboration from Amar, Italy, and we got a foundation from Coral Reef Monitoring Program from World Bank, and this all of the people in my group. So we are young researcher. So some of them PhD, some of them still masters, and want to pursue the PhD right now. So if you want interesting to join our lab, we in LIPI right now, we have the program, some of postdoc or visiting researchers. So please come if you're interested and contact me. Thank you very much.
All right. Thank you very much, Dr. Mastaria.
I can drink right now.
That was such an interesting take on marine natural product. I never thought that it would be. I never thought that it would be that much species of sea cucumbers. Actually,
yeah,
that was over 20 of them. That
Sorry?
was so many of them.
Yeah. We collected it.
When you're exploring this marine species, how often did you found a new when you are doing this? How often did you found a new species?
Often I found of new species. Yes, I can hear you. It's difficult to find a new species, to be honest, because most of them are already identification. But I always go to diving with taxonomies, because if you're working with marine natural products, you have a close collaboration with taxonomies. So that's very important. So I have a good friend and have a good collaborator also in DP. So he is he is the taxonomist for sponge. So but he understands also with soft coral and Shidian. Sometime we predicted new species, right? But right now, if the new species, we need to combine also with the classical taxonomies, mean that you have to understand the spicules of the type of the spicules of sponge or soft coral, and you have to do the molecular taxonomies also. So you do have to do DNA extraction, something like that, and need to understand the molecular taxonomies there. And I think we have one or two right now the potential for new compounds, but not from sponge, but from the coral, hard coral, because most of my collaborator also marine taxonomies also in hard coral and in sponge. So, but in hard coral we cannot study on marine natural product because the concentration with the calcium carbonate. So, but we isolated marine microorganism from the hard coral also.
Where did you find most of this, soft coral or the sea cucumbers?
Mostly in East Indonesia we collected. So we tried couple of times diving in West Indonesia. We tried a couple of times diving in Nias. We tried a couple of times diving in like Mentawai, Sabang, and Bangka Belitung, and Anambas. We have been there also. But to produce the natural product, I think still is Indonesia still interests, because most of pundan of the marine organism there.
Yeah, so if you want to look for something that is not that much explored, just go to east part of Indonesia, something like that, right?
Depends, but we are also interesting with in Jakarta also. We are also interesting with Seribu Island, because more pollutant. So Jakarta is very pollutant. So if the extreme condition, people organism can produce, then maybe the other molecule. That's why I mentioned before. Different condition. That's why interesting with marine organism. In different condition, they can produce the different molecule. So we collected the same species in the same place, but we also looking also the environmental. That's what I said to you before. We collected also in the volcanic sea in Sangihe, also in Aceh, but we collected the same species, but they produce sometimes different molecules, not the same. The major molecules may be the same, but the minor molecules different. So they make the new adaptation with the different environments.
I see. So basically, this is like evolution in action, basically.
Yeah,
So if you
evolution for the
found
extreme
the same
condition.
in different place, you will found two different things, something like that. At least in the details. All right, Dr. Mastariat, thank you for your amazing presentation. But first, before we are entering the Q&A session, we first take a break and watch some nice video just to cool off our brain so we don't overheat our head. And for participants who have any question regarding the lectures, you can put your question in the AHA slide in the chat room, or you can just raise hand and we'll allow you to speak directly to Dr. Masteria. Thank you. Alright, that was some video break, so we don't overheat our head, hopefully. But don't get your head too cool, because perhaps some of you have question that you would like to ask to our lecturer in this session. If you have any question, you can raise your hand. Safai Hassal, perhaps your mic is open. All right, I think we should we'll go to the AHA slide to see some of the questions that have been posted there. Who will share that? I think it's Pak Abraham. Is it Pak Abraham? All right. Okay, this is some of the questions that have been asked in the AHA slide. First is, what is the main motive or skeleton of natural products that make them different with natural products derived from higher plants?
I answer directly, or
this is a bit strange. Yeah, you can answer directly.
Okay.
Please,
thank you,
Dr. Masteria.
Yeah, what are the main motif of skeleton marine natural product differ from natural product different from the higher plants? Difference, yeah, difference, because mostly in terrestrial is plants, right? Terrestrial plants, but in marine environments, most of them animals, invertebrates, and most of them microorganisms. So that's kind of difference of type of macroorganisms. And the others is the marine organisms. They are filter feeders, and also because that I mentioned before, sometimes they eat for the growing, they eat bacteria or microalgae or something, and something that come to the their body, something like that. So that's the difference, both of them. And this is also continue the second the question number 1 and number 2. I think I can answer together. So what is the more interesting for the new bioactive right now? In the past 30 years ago, 40 years ago, we not interesting test from the stress ecosystem, damage ecosystem, or unpollutant or something like that. Something we're not interesting. We interesting. Okay, we collected so marine organism, but now researchers is understand that in the marine in the marine environments, the ecosystem is very important to produce the bioactive molecules. Because what? Because why? Because that's what I say, because they are not moving. So there is a competition space based on them, and in the coral reef, and there is also their filter feeders. So different bacteria that they eat, different bacteria in surrounding them. So right now we interesting with the extreme conditions, because we believe with the extreme condition we are different adaptation and different produce the new molecules. Even we also interesting with the organism in the deep ocean also. So you can see most 5 years in the past 5 years there's several paper mention about marine natural product from the deep oceans. So number 3, what you mean domesticated the invertebrates? How effective will it be? I don't know what you mean domesticated here. Can you explain it?
Let's see. I think what he meant by domesticate is something like, can you perhaps make it like a fish? So you just make a place for a fish, something like that, I think.
Oh, you make the aquaculture or marine culture, something like that.
Perhaps.
So,
I'm not sure.
I said for the marine culture, or what you say, domesticate, I think not very effective. For examples, in my presentation, that what I mentioned, I present about the plitidepsin here. So plitidepsin from the Ascidian Echinacea turbinata. So they produce the plitidepsin that for the anti-cancer drugs. So the first thing for the market, they produce with marine culture. So they make the aquaculture for the Echinacea turbinata. but the problem is sometimes the condition is a difference, the environment's difference, and only 1 milligram from 1 kilogram frozen or from wet ascidian. So it means that 1 kilogram only produce 1 milligram. So that's not interesting for market or for drug system. So they need to do the semi-synthesized or synthesized with the cheapest step.
Basically it's a bad business idea.
Yep. And second, what kind of marine?
I have a question, Dr. Mastaria. Can I ask a question first? Yeah, you mentioned before that now people also interested with something marine product from the deep sea, right? So have anyone extracted any natural product from the organism from the deep sea fence?
Deep sea fence, yeah, several here, and also we have also the collection from deep sea. So, but we still not focusing about that, about what we deep sea, because we have collaboration with China. So we did one expedition last year with because in Lipi we have the research center for deep sea. So they did the expedition with China, and they give us some of the samples there, but we still not doing. We still not continue the process, but we want to continue in the next years maybe. So it depends on the money that we have it. But in some of the peoples right now in Japan, because in Japan so many people working manner not natural in the past 20 years ago, in the past 10 years ago also, and mostly sponsors in Japan they already do the extraction, already publish everything, but now people in Japan interesting with the deep sea organism and also with these deep sea microorganism. So you can find in the paper like marine drugs, journal organic chemistry, journal natural product about deep sea natural products.
I see. I was a bit interested because as far as I know, if near the deep sea fence, right, when we are talking about ecosystem that is near the surface and in the deep sea, they are very different. When we are near the surface, the bottom of the food pyramid is basically plants, right? Basically, organism that can do photosynthesis. But when you are going to the deep sea, most of the bottom of the food pyramid is organism that do chemosynthesis, which is a bit different from photosynthesis. That's actually very interesting to me.
For sure, that's interesting. They have the chemosynthesis.
Yeah, I'm honestly, so I'm actually really interested how do chemosynthesis and photosynthesis would they produce different whether primary or secondary metabolites, and if they're different, how are they different, something like that. Do you have any data on that, perhaps?
We don't have it, but that could be depend on the environmental. That's very important. So that's what unique for the marine environments, a marine organism. Different environment, they could produce different molecules. So we cannot compare with one species that we collect in different area. They could have the new molecules. So that's what I said. Mentioned it before. The environments, that's very important. So you have to know the condition of the environments where you collected the organisms.
I see. So we also have question here from Unita. Unita, are you here? Hello, Unita.
How to good preparation sample to get mannequin.
We have Unita Purnamasari over here.
So I can answer.
Yeah, please. Please.
Okay. So how much minimum marine sample amount to isolates? That's very tricky because why? We cannot collect all of them. We cannot collect all of them because we need also to aware about the environments. So if you collect the sponge or ascidian, don't collect everything. So just cut off them and remain half. That's very important to maintain of the biodiversity. So for me, we only collected around 500 gram samples, or just one of like my hand here. So this is the maximum that I collected for the marine sample. I don't collected 1 kilogram, 2 kilogram of everything. I just collected here. Like why I don't know in English here, but only here in my hand. I never collected more than that because we need to maintain the biodiversity.
The size of your fish, that is enough.
Yeah, this is enough for me. This is enough for us.
Wow.
This is enough for us.
I mean,
this is enough. That's why, because
I mean,
that's why the challenging for to get the new molecule in marine organisms. You cannot collect it all of them. You cannot collect 1 kilogram, 2 kilogram, 3 kilogram because you have to maintain biodiversity. You have to be maintain the biodiversity. You cannot lose it sponge because sponge can be growing 10 years, 2 years, 20 years, 30 years. So only for the marine natural products, that's not we don't know that's only being only a paper or can be being in the market, but we lose the biodiversity, and that's not I think to compare, that's not a good idea. So that's why in most of the marine natural product chemists, they never collected all of the sample, believe me, because they should be maintained biodiversity. So how to good preparation sample to get many kind of compounds? So first, there should be the fresh material. So if you go to the collection, just prepare the dry ice or something like that. And after you get to be in the mainland, so just put in the freezer -20. That's very important because there could be degradations of the compound. So should be put in the dry ice first, or just put, or if you don't have any dry ice or you don't have any in your place that where you collected, you don't have the -20. I think one of the suggestions that I want to mention, just put the alcohol. That will be okay. Just put the alcohol. So if you don't have dry ice, just put the alcohol. Yeah. Because this you can prevent from the smell, very smell, because very smell, very nasty. Then and also you can maintain your organic compounds. But my suggestion, just the first suggestion, find dry ice. If you don't have dry ice, yeah, just put alcohol, something like that.
I see that is a very interesting suggestion. I've never thought about it like that. I mean, I know that alcohol can be used as preservative, but not like that. I never thought that you can use it to preserve marine products.
Yeah, that's the last option. That's the last option, because if you don't put alcohol there, very smell. The smell's not good, and people will be angry to you when you bringing when you go by airplanes. The people will be angry to you. So that's my suggestion. But the important thing, using dry ice.
Okay, so we have next question. Wait, in what kind of marine ecosystem is more promising to live? Yeah, I think you have answered that one. Together with the first, dear sir, what are the anti-cancer activity tests that are low cost but acceptable for international publication. As I know, anti-cancer activity tests using cell line are so high cost. Yeah, yeah, I know.
Yes, high cost.
So, any suggestion?
Very high cost. I believe that very high cost. You have to get great grants. So, for the simple cytotoxic using the brain stream lethality assay, that's the simple way. So not very cheap. But if you want to understand the cytotoxic with the cell, you have to do the MTT, also sulforadamine. But for me, with much more expensive sulforadamine or MTT, I think the most I prefer the sulforadamine B, because in our calculation that's cheaper than MTT for us. And yeah, for the maintenance of cells, very expensive also, but yeah, just doing one cells, maybe that's really interesting for me. If you want, for starting for the cytotoxic activity, for me, I'm very interested in using breast cancer, because in my experience, if they're active for the breast cancer, they will be active for lung cancer, they will be active also for the colon cancer. But if they're active for the colon cancer or lung cancer, sometimes they're not active for the breast cancer. So for me, the starting point for me is breast cancer, because different type of the like metastatic, different morphology of the cancer. I'm not more expertise with the cancer, but breast cancer is more difficult than lung cancer and the colon cancer for the inhibition. So just doing just one, or just use one breast cancer, I think, for the first step. If you're interested to find the new active molecule for colon cancer, for the activity for the lung cancer, we can do the next step. That's the simple answer from me.
Yeah, yeah. I realized that for the breast cancer, you use MDA cell line.
MDA and B231s, but sometimes there is the MCF1.
Yeah.
MCF7s.
Yeah,
and
yeah,
what else?
Most of the time MCF.
Yeah, MCF7. Yeah, different morphology, but I think MDA more difficult to inhibition than MCF7, in my opinion. That's why I prefer to use the MDA. And also in my previous lab in US, because I studied the anti-cancer mechanism of anti-cancer in US, they use the MDA more than MCF7. That's why I prefer the MDA.
Right. First, I would like to before we continue this, I would like to invite the participants to perhaps ask directly to Dr. Mastaria. Does anyone have any question that they would like to ask directly to Dr. Mastaria? Please raise your hand. Hello? I don't think they have a
question.
These people are too shy. So they just write the question. Oh, Wendell.
Hello, good afternoon.
Good afternoon.
Yeah, I have a question about sponges a while ago. Why are sponges important sources of marine natural products?
Sorry, can you explain it? Repeat it.
Why are sponges important sources of marine natural products? I am curious about the sponges.
Yeah, that's very interesting. Why the sponge is very important for marine natural product.
Yes.
So first, they are more abundant species than the others organisms in coral reef, and the second is much more easy to collect it than Ascidian and Tunicat, because the Tunicats is very small, Ascidian is very small. Only Polycarpa is more the same bigger with the sponsor. If you collected Ascidian, the Ascidian they are collected only here, sometimes only here. So you have to collect it more Ascidian or Tunicat in the same area. Okay, you can collect it in different area, but in the same area in 1 meter or 2 meter, because more than that is different condition. But the second part mentioned that they are the abundant, and in the abundance species in coral reef, and the third part is they are bigger, more bigger than the others organisms in coral reef. So that's why I mentioned before, if you want to collect the tunicats, you have to collect it several times. But if you collected the sponge, they are bigger. Here, you just cut it. So like you see in the video, there's the big sponge that's named the Schistospongia, right? And you just collected small part of them to study the marine natural product. And also soft coral, I mentioned two to three of them, soft coral and tunicate, right? And soft coral also, they are the same big as, if compared the bigger, it's almost the same with sponge, but soft coral, they're not much more species than the sponge. So I think soft coral only, I think the most important species 5 or 6, or 5 or 6, like Sunularia, Sarcoviton, something like that, or Xenia, and sometimes difference in different area. And other things is sponge, they can make the adaptation in the extreme conditions. Then the others, tends the soft coral. Soft coral, they cannot growing in the more pollutant, as I know. They cannot growing in the pollutant area. And also as Ascidian, they can grow in the pollutant area, but they are very tiny. That's why people, most of the publication marine natural product came from sponge.
Thank you, sir.
Thank you very much. Welcome.
Well, that is a very interesting take. Oh, we also have another question here. Pak Abraham Mora, please.
Okay. Can you hear me?
Yes, we can hear you.
Okay. Good afternoon, Dr. Masteria. I'm Abraham, a part of the committee, but I have a question because I'm so curious about your research. The question is how you collect the sample, maybe how the technique to collect the sample, because if you don't not careful in collecting the sample, it can be harm for all the ecosystem, right? It can ruin the ecosystem. How can you make sure that a collection sample collection is safe for all the ecosystem among the marine? Thank you so much.
That's very nice question. That's what I said, how to maintain the ecosystem. So you maintain the ecosystem, you maintain the biodiversity. That's very important. First, you have to able to diving. So not many people, mostly people only recreational diving, right?
Yeah.
So to see the coral reef, something like that. But in the area, if you are researchers interesting with the marine or ocean, you have to know knowledge how to do a good scientific diving. That's very important. So if you know the scientific diving, you know how to be aware with the marine ecosystem. For example, when you use your finch, right? You finch when you do the diving finch, and you can kick some of the marine organisms also.
Yeah.
And that's depend on the people, I think, and that's depend on the skill of the people to do diving. for me, I maintain the ecosystem. I only go to diving first in the deepest area that I want.
Okay.
So not from the like from 1 meter going to do the 40 meter, but for me, I will go to the 40 meter first. Okay, jump to the 40 meter first, and you start to collect the sample, because when you collect it 1 meter to 2 meter, your fins will kick some of the areas, some of the organism very easy, right? But when you kick, you just could jump out first, then not much or organism that you attach first, and that's what's our technique for doing the diving. The second, you have to bring more like net that you use it. So like for example, that I use it here when I collected sample, right? Let me show to you.
Yeah.
So that's it. So use net here and use fins here. So here the sample you collected here. So don't maintain here. I never leave my net to everywhere. So I'll touch my net in my hands always. So because net could be damaged also the other organisms. So that's it. So don't touch anything. Use your if you have the glove, use glove. So because sometimes you can get hit, sometimes you get some of toxic compound that produced from the marine organism that make your skin burn, something like that. So that's the technique that we collected. And the second part, you have a body. So you have a body to diving, your friends, your college, your dive buddy that can be your partner, because you go to diving, yeah, you go to the deepest area. There is something can be happen there. You can find shark, you can find snake, right? So to protect you or to make you aware, you have to buddy. Don't never go to diving alone. That's what our techniques. So they will be advisor for you. They will be remind you something that you forget. That's what our techniques. So first you have to do scientific diving, understand the scientific diving. That's my answer. Abraham.
For instance, I have experience to snorkeling in the Pulau Seribu, and when I touch the corals, I feel that the corals is very sharp, and I don't want to touch it anymore. My curiosity is how you such as cutting the corals. Did you just take it, or you kind of cut it first, or just using your net? I still have no idea about that.
We use knife. We use knife.
You use knife.
Yeah, we use knife. So we never take it by hands. We use knife.
Okay.
We use knife. Sometimes we I don't know in English, we bring some like I don't know in English also in English. But okay, mostly we use knife. We use knife. We use knife to touch. We touch with knife first, not with our hands. So because even you use glove, you do not touch is toxic, right? Then then will be make your hands damage or something. But to understand first, we use knife to touch first.
Okay, my last question maybe Pak Hafiz, can I?
Yes, please.
Okay, so what is the most danger or challenge that you have while you collecting all the marine sample?
The most challenging is snakes. Sea snakes.
Oh, really? You
That's
have
the most challenging.
snakes.
Sea snake is very poisonous. I think the very poisonous snake in the world is sea snake. So that's the most challenge.
And how you are dealing with snakes?
Calm. If you find if you find snakes, just calm and go to the other area.
Yeah. Okay. Thank you so much for explanation,
Thank you.
Dr.
Thank you.
Mastaria.
Okay, thank you, Pak Abraham, for your question. Dr. Mastaria, I think you just get yourself a budding marine chemist there who is ready to dive. Hopefully,
he can come to my lab.
Yeah, I'm also, even though I'm not I cannot swim, I'm actually I do know that people can dive without even swim without knowing how to swim, because as far as I know, the buoyant force actually can sustain us quite well in the seawater, right? Because
to get certificate,
of
you have to
the
able to
higher.
swim. To get certificate, you have to able to swim. But the
I
recreational
see.
diving, you don't need to swim.
Oh,
You don't need to able to swim. Just go to Bali
yeah,
and
I
find
see. So if
find
I
dive
want
center.
to
You can help. They can help
just
you.
dive for fun, so yeah.
Yeah,
So if I want to dive just for fun, so I don't need it to know how to swim. But if I want to get a certificate, I have to
swim.
I, I think I need to learn to swim. Okay, I think we can go back to let's see. I want to see the. Oh, we have Arif here. Arif, are you there? Arif? He put his
yes
question there. Yeah, perhaps you can ask your question directly to Dr. Mastaria.
Okay, thank you, Mr. Horis. Okay, good afternoon, Bapak Mastaria. Thank you for such insightful presentation today. I'm sorry I cannot edit my video because my internet connection is slow. So my question is about I'm very interested with the natural product, and based on your presentation, you explained about your research before about sea cucumber. So I want to ask about the cucumber. What makes sea cucumber very interesting to or to bring this sea cucumber to source of marine natural or marine drugs than other microorganism or other marine microorganism. And right now, what does the sea cucumber product that has been published in industrial? Thank you.
Thank you. Thank you very much, Arif. Nice question. Yeah. So why we are interested with sea cucumber? Sea cucumbers, one of the, we call it the ginseng sea. So that's what we call it, the ginseng sea. So most people from China, Korea, in East Asians prefer to eat sea cucumber because they believe that it's the same effect with the ginseng. But anyway, why I'm interesting is because in Indonesia, that sea cucumber have been over exploitation. So most people in East Indonesia collect the sea cucumber and tried it and export it to the, like that I mentioned before, China, Thailand, China, mostly in China. And they have the activity also first for the anti-cancer, second, and that's so if you're looking for the scientific paper, so many publications about the saponin from sea cucumber have the potential for the anti-cancer. And the challenge is how to develop this sea cucumber to be a market in the drugs, such as we call it is of not only not compounds but only the extract that maybe can be do in the market also. So that's why we collected all of them. So we understand we want to understand the chemotaxonomies. We want to understand of the molecular action from this cucumber, even the extracts, and we want to develop this to be the drugs here, or maybe we call it herbal, the first herbal from the sea. That's our main goal. And one of our collaborator from the KKP Ministry of Fisheries already make the marine culture from the sea cucumber, two type of sea cucumber, Holoturia scabra and Stichopus hermanni. And the Stichopus hermanni, right now, they have been in the market. We call it gamats from Malaysia. So mostly the gamat from the Malaysia, that they call it gamat from Malaysia, mostly came from Indonesia, from the Stichopus hermanni. And now people, one of the collaborators from the Ministry of Fishery already able this marine culture for the from the Stichopus hermanni. And that's why we're interesting. If we can do a marine culture of one species, we can do the marine culture of another species. But we need to understand it, the activities this cucumber first before we do the marine culture. If we're doing the marine culture but they don't have interesting pharmacological activities or biological activities, or they're only interesting only for the functional foods, at least we can do it for the marine culture. That's why we're interesting for the sea cucumber. Mostly sea cucumber here, mostly sea cucumber functional. We develop functional food, but right now we have the funding from the Indonesian government to develop it sea cucumber as the herbal right now for the anti-cancer. So for the adjuvant for the cancer, that's what our dream for the sea cucumber, Arif. So this is my answer. Thank you. Maybe.
Okay. Yeah, thank you, Mr. For the good answer. And because I also have joined some of the previous like virtual talk about marine that you become the speaker. So we have in Indonesian in LIPI also have like Burati that bring industrial in seaweed, if I'm not wrong. So I think this sea cucumber also can produce or bring for in Indonesia. Thank you so much. And also I have
seaweed is for functional food, very interesting, but not interesting for drugs.
Oh, for drugs.
So functional food is interesting, but to develop seaweed for drugs is for me, for a simple question for me, it's much more difficult, because mostly seaweed is fatty acid, mostly the content of the seaweeds. But if you compare the sea cucumber, they have plenty of the saponin that can be produced for the drugs. That's very interesting right now.
Okay, thank you. And also you mentioned before that if have like program like visiting research or postdoc research in your laboratory, so maybe we all the participants in this summer is very interested with that program. So maybe please give up in advance information about this. Thank you so much.
You can check in LIPI. There is called the Pusbindiklat LIPI. So there is the visiting program in the Pusbindiklat LIPI for the bio research. So just check in the website of Pusbindiklat LIPI. I can write here. So you can find more information about the visiting researcher or research assistant or postdoctoral in LIPI that you can apply for this research expertise that we want.
Okay, thank you. I will check. Thank you so much, Dr. Masteria.
Right. Thank you, Arif. Perhaps do you have any other question, Arif?
No, I think that's enough. Thank you so much.
All right. Thank you. Okay. Does anyone else have a question that they would like to ask to Dr. Nastaria? Please raise your hand. Okay, I think we would, while we're waiting, we will back to our slides over here. So here is a question. Dear Dr. Mastaria, thank you for your lecture. I have some simple question. When you're exploring marine natural product, how often did you find a completely novel compound?
Completely novel compounds, it means the skeleton of the natural products, right? The skeleton, the new skeleton of the compounds. That's very difficult. That's interesting question, but difficult to find the completely novel compounds. Mostly the novel compounds that right now in marine natural product or even in terrestrial natural product, they are only different with the different stereochemistry, different attached additional ring that they have it. But the completely novel compounds, I think it's difficult to find it. Even that mostly that I collect isolated novel compound that we saw isolated is, I think, only that the alkaloid that I mentioned before, but that is still contain the fatty acid ring additional. So complete novel compounds is difficult for me. I mentioned, to be honest, it's difficult to get it, to be honest.
So very, very rare.
Yes.
Yeah, actually, regarding this question, I actually have a follow-up regarding this question. When you did found them, how do you even identify them, right? Because as you know, usually natural product tend to be very complicated in its structure, and if you are using just proton NMR, I think the signal would be just a mess. So it would be very difficult to elucidate what that compound actually is. How do you even identify that whether you have a new compound or not?
So first, MS. So we need to do the replication, search in database. If we believe it in database, that's the new compound, even with stereochemistry. So you need to do NMR. That's the basic thing that you have to do. I think you have to do 2-dimensional NMR, like COSI, TOCSI, SAQC, SMBC, something like that. So you have to understand it. If you want to do the absolute configuration, you have to do DFT calculation, ACD spectra, or X-ray crystallography, something like. So yeah, much thing we need to do, and much thing we need to learn about the structure elucidation. But the important things that if you already understand b-dimensional, that's enough for you to structure elucidation. But to configurational, that's what I said, you have to do some FDFT calculation, compositional study, something like that.
I see. So because it is very, we don't know whether it's real or not. We just have to check at first using simulation, right?
Yeah, but if you're
using
confident,
computational
but as I
chemistry.
mentioned before, if you in confidence in database, not much wasting time. If you believe that's new compound and database mentioned that there is no record about your molecular weight in database with the molecular weight in the same species, they don't have it. So just do NMR. I think much more COSI first, or NSCQC, COSI and SMBC first. If you already got it, doing the stereochemistry with CQC, something like that. So much more important thing, do database first, not to because if you do database, you're not wasting your time.
I see. All right. I think that settles the question. Right. The next question. Does the depth of the ocean can affect the characteristic or complexity of the secondary metabolite structure produced by the marine creatures?
Yes, because there are different environments and there are different adaptation, and maybe there are different bacteria inside there. So deep ocean is very interesting right now for to get the new molecule.
I see. So it is the simplest answer is yes, it can, but further information you have to check other reference. Okay, the next question. I'm not sure we can consider this one a question. It's more like a personal one. Oh, now there is another one. This one is from Nilofar. Nilofar, can you hear me? Hello, Nilofar? Yeah.
That reliable database,
yes.
that free. That's why I'm still looking also for that. That's free because very expensive.
Right.
Database is very expensive. From Lit, maybe I think 10,000 euro or something like that. That we need to get the access for the database like Marine needs. So to be honest, I still don't get it. The free database that really comprehensive like MarineLit or CFinder, right? So to be honest, no.
I see. So the answer is not yet. So we have not yet found one that is free and comprehensive. Let's see. Oh, here's so he actually she actually further add to her question. I mean, can you suggest some website that are free to have some look? I think the best one we have is SDBS. Is the best one we have that are free. But you know, SDBS is not that comprehensive.
Yeah. So there's a question from Unita here. Do you also isolate microorganisms?
Yeah.
Yes, we isolate also the microorganism. Then how to ensure that isolated microbes are really isolated from marine organism, are not from water? So this is tricky. So that's very tricky to isolate marine microorganism from the marine environment. First, you collect the sediments. The second test is from the symbiotic from a sponge. If you check it from the water, that could be from your sponge. The other thing that if you collected most of this like a sponge, they are symbiotic with the nudibranchs, the tiny animals that show in the first video. So mostly ascidians have symbiotic with the sponge, and both of them eat the same microorganisms. So if you collected the same microorganism with nudibranchs and the same microorganism, the sponge could be test the symbiotic microorganism in sponge. That's something that I can explain to you based on my experience. So check it in your sponge, check it in your sediments, and check it in your water. If you want to understand, that's really from your organisms.
I see. So basically just check it in multiple places just to confirm whether it's come from directly just one place, right? Okay. I think the question from Nilova is already answered. This one I'm not too sure. In your graduate studies, you were working on isolation, and now you're doing synthesis as well. Am I correct?
Okay.
I think,
so
yeah, I
that's
think.
Can I
Yeah,
answer?
please.
Yes. So I did
Yeah,
semi-synthesized, not full synthesize. Okay, not total synthesize. So I only understand with semi-synthesize. So total synthesize, not my experience, and not I don't have background with synthesize. So but simple synthesize, I can understand it. And the second part, I explained my studies to medicinal chemistry. So that's why I learned about the anti-cancer mechanism of action, something like that, when I start my postdoc in US. So that's why I mentioned to you, my background is compared with the chemistry, biologists, and biotechnologists and pharmacists. So because my PhD, my bachelor degree in chemistry, my master and my PhD about marine biotechnology, or my research about natural product chemistry. And my first postdoc in Germany, I working about the marine ecology, chemistry, ecological chemistry, from sponge and from soft coral or tunicate, something like that, but in area of microorganism. Then when I understand the chemistry and biology, something that with ecologists, I really interesting also, because if you compare with natural products, you have to understand biologics. You have to understand biological activities. You can understand how this part of the compound that you isolated, maybe there will be activity with some of the biological activities. That's why I'm interesting to continue my PhD, my postdoc in medicinal chemistry. So that's I combine in several area expertise. Hopefully I understand. Hopefully I understand everything, but yeah, I'm still study everything also.
All right, that question. Wonder how to do sampling and how to keep the sample from any change.
I think I already.
I think you just have to.
I think I already
yeah,
answered.
I think you have answered that question. Yeah. The next question: based on your experience, where the place in Indonesia or other that have so many unique marine creatures?
Many creatures. Have so many unique creatures. Oh.
That's a difficult one.
I think in Buton.
Buton.
Buton Islands. In the Buton Islands, because there are different environments and different in terms of the environments, not pollutant. Yeah, in terms of the environments of surrounding with wave, extreme conditions, and geographical situation. So some of the other parts could be different major component of organism. In some place there is so many soft coral there, and the other place so many spawns. That's very uniqueness for me in the same islands.
I see. Okay, next. Are there any phenol compounds found from marines besides algae and seaweed?
There is some of the phenol compounds, but not much more phenolic compounds, I think, because phenolic compounds mostly in terrestrial, right, from plants. Some of the pepper there is phenolic compounds. Some of spawns there is there, but from soft coral I believe there is no phenolic compound, because most of the soft coral only sterol and diterpenoids, and some of them glycolipid and alkaloid. And from sponge, I believe, yeah, there is some of the phenolic compound have been isolated there. And from the ascidians, no, I think. From cyanobacteria, I think they don't have the phenol compound. Mostly cyanobacteria has peptide compounds. So yeah, that's it. I think maybe we can you can search in sponge.
I see. All right. Next question is, another one from Nilofar. I'm trying to isolate alkaloid from plants. Should I use silica gel column or something other to suggestion for separation for alkaloids?
Okay, plant is not my expertise the area. Yeah, you can do the silica gel column, or maybe yeah, I think maybe you need alkaloids. Mostly the alcohol is polar, right? So yeah, you can do silica gel column, or you can do other methods, maybe like gel column chromatography, counter current chromatography. What else you want to do? But for me, I'm basic not only to search the alcohol, some of the particulars compounds, because for me I interesting all of the type of the compounds that have the biological activities. That's for me. So I'm not interested in one type of the compounds. So I just can explain to you in traditional methods. So just doing the extraction, do the silica gel column, collect your fraction, or do some of the mass spectrometry, that if you have the targeted for the alkaloid, so you know that's contained in your fraction, something like that, and do the NMR. So this is my suggestion, Nilofar.
Okay, and next, what is the most challenging step when you isolate secondary metabolites from marine? As far as I know, the source of marine is very little.
Degradation of the compounds. That's very challenge. That's a very challenge, because mostly they have the double bonds, so it cannot be stable condition. And what else? If we collect the marine organism, when we do the extraction, the colors very dark. I think sometimes there is the color with red, sometimes the color with yellow, sometimes with color with brown, something like that, and could be possible we can get degradation of the compounds. So that's very stable. That's a very challenge there. And also for me, when I do the extraction, I never put my sample near the suns or near the light. So I always cover them with aluminum foil and put them in laminar air cabinet, something like that. So that's the most challenging.
Right. Next, special condition to store fresh sample. I think you have answered this one using dry ice, or in the last resort, alcohol, I believe. Okay, do you think it's possible to apply bioactive compound from marine organism besides pharmacy, such as FNB?
Food and beverage. For sure, yes. We have microalgae, and microalgae that
Food
right
and
now
beverage.
using for food and beverage. So pigment from macroalgae use the food and beverage now. But be attention, some of the macroalgae there is also from fresh water and also from the sea. So be careful about that. So, but what I said to you before, yeah, it could be possible from the pigment of the macroalgae or from the pigment from algae.
Okay, we have one question here in the chat room. What is the main difference between fish and crab in terms of nutrition. Which one is more healthy?
Fish and
crab. You know,
if you're
the
asking me
crustacean.
fish, if you're asking me fish, the crabs, if you eat, you'll get cholesterol. So, and you get a hypertension. So, fish, you can be smart with this fish.
So, fish over crab, right?
Fish, for me, fish.
Okay. Okay. Next, I think we can see from when you investigate sponge, did you extract the whole part of the sponge or just pick some part? I think you have answered this one. You just pick some part of it and extract from that. All right, and the next one, I read in your PowerPoint and that one chemical compound that we can get is pigment. What is the advantage of the pigment?
What is the advantage of the pigment? So I think most of the pigment have the antioxidant activities. Most of them, like fucoxanthin, astaxanthin, or what else? They have the antioxidant activity. So that's one of the advantage for the pigment. One of them for the color of the pigments could be natural. So it be healthy for us to drink or to eat the pigment from natural.
Environmentally friendly. Yeah.
Yeah.
Okay. The two question from anonymous is already answered. The last one from Nilovan again about plants. If I have to work on your field, what kind of challenge I need to face? I mean different from plants to this kind of material in terms of technique only. So she asked if she want to switch from terrestrial plants to marine product, what would be the challenge?
The challenge is the collection. For the techniques, I think most of the technique from if you're working with terrestrial natural product or marine natural product, the same. You did the extraction, you did spectroscopy, such as LCMS or NMR. That's the basic thing you need to know. You have to do, if you want to more experience, you did the biological assay, like screening, it's almost the same to know. But the challenge is collections. So if you're working with plants, you can collect easy. You can go to the forest and collect the sample. But if you challenge, if you work with the marine, you have to know first. If you cannot do diving, you can do snorkeling. So that's you have to do swimming. Swim. You have to able to swim. So that's the challenge. I think in terms of the technique, almost the same. Almost the same.
I see. So basically, in terms of technique, basically the same. It's just that if you want to get at your sample, you have to be able to swim, something like that. So
or
if
you have a partner, a college that can do a diving.
Yeah. So either you have to get it yourself or get someone to get it for you.
Yes.
All right. I think that's all from Nilofar. I think that's the last.
Thank you very much, Nilofar.
Yeah. All right. Thank you very much, everybody, for your question. This is a quite intense Q&A session because you have like dozens of questions, some of them related to each other. Some of them is very interesting, like what kind of biodiversity how do biodiversity can be affected by the environment and the effect on the chemistry, and as well as if you have the sample, how do you keep it fresh. So thank you, Dr. Masteria, for your amazing lectures. I really hope that we can speak again sometime in the future. we have a very intense discussion this time. As a token of our appreciation, this is a certificate for you. We will send it to your email directly as soon as possible. And before we are closing, perhaps Dr. Masteria, you have a closing statement.
Thank you. Yes, thank you very much, Dr. Hafiz. Thank you very much for all the organizing committee for the invitation and for the certificates. If you have any question for me, if you want, we can. For in our lab, we are open to collaboration with students, any other researcher from around Indonesia or from around Southeast Asians. So please, if you want interesting, please contact me by email. So I think you already have my email, right, from the first presentation. So
yes,
see you
yes.
in the next events. And yeah, I hope you can join. Yeah.
Yes.
Thank you very much.
Thank you very much, Dr. Masteria.
Yeah.
Thank you very much. So thank you very much to Dr. Masteria for his amazing lecture. Dr. Masteria, if you have any other activities after this, you are free to leave this meeting. But if you don't mind to stay, you can and still join us until the end of today's session. After this, we have a quiz. We have a photo session first, right? I forgot about that. First, we have a photo session. Everybody, first, please open your camera. Okay, Ibu Tri Anissa, I believe, would be the one who take the pictures.
Okay, thank you very much, Pak Hafiz, and also thank you very much, Dr. Masteria, for giving your very nice presentation. And so, ladies and gentlemen, this is the time for photo session. So, for everyone who has any trouble with your connection, please open your video so we can get the picture. Okay, I will count in 3, 2, 1. For first slide, and then continue for the second slide. 1, 2, 3, and then for the third slide, and 1, 2, 3. For the fourth slide, and then 1, 2, 3, and for the final slide, 1, 2, 3. Okay, thank you very much. This is very nice presentation of Dr. Masteria. So I give it back to moderator.
Thank you.
Thank you, Ibu Tri Anissa. Now, after this, we are moving to the quiz session.
Thank you very much, all. So I can leave it, right?
Yes.
So thank you.
Thank you, Pak Masteria.
Yeah.
That
Thank
was
you
a
very
very
much. Bye-bye.
interesting discussion.
Thank you. Thank you. Thank you. Bye.
Thank you.
Bye.
Yes. Okay, this is a quiz time. Okay. I have just sent you the link to the quiz on the chat room. You can also see the link to the quiz on your monitor right there. ahaslides.com/vss9quiz. Please click the link and join the quiz. So we are still waiting for players to join. people who have not yet joined there, 70. We'll start at least 80. So come on, please join us at this quiz. $75. 76. How many participants that have not yet joined here? Come on, we still have sometimes. Please join on ahaslides.com/VSS9 quiz. How many? Come on, everybody. Don't be shy, just join. 79. Okay, one more. Come on. Just one more. Why did it decrease? Oh, it's actually, it decreased even more. Why is that? All right. 78, 79. Come on. Just one more players. All right. I think we should just start. Who is in control? Okay. Pak Abraham, please start the countdown.
Okay, sure. Okay, so guys, we're going to start our quiz. Yeah, so prepare yourself, and the quiz will be start in 3, 2, 1, and good luck.
All right. 31 participants answered the question correctly. In the first place, we have Nabila Soraya with 90 points and Dian in the second place with 83 points. All right. Let's see to the next question. All right. Second question. The following are sources of marine natural products for application in human health except sponges, corals, high plants, actinomycetes. Alright, which one is it? And the answer, of course. Is high plants, obviously. Let's see who is on the leaderboard right now. Oh, Nabila Soraya, keep her lead. She's now 19 points over her over the runner-up. Dian Graheni is overtaken by Dian M. Now here's a structure of the compound and what activity does this compound have? Time's up, and the answer is anti-cancer. So most of the people actually answer it correctly. Let's see the leaderboard. Nabila Suraya kept her lead at 249 points in front of Dian Musdalifah with 219 points. All right, let's go to the next question. So, which compound is not the main feature of compound isolated from soft corals? They are in a ring, oxygenated, rigid, or flexible? Well, obviously, okay. The next question. Well, first we see the leaderboard first. Oh, Nuna Zatun actually overtake Nabila Suraya. She is now 30 points up in front of Nabila. Let's see the next question. Can Nabila reclaim her position? Toxicity study. All right, how many of you answered correctly? Oh, we have a newcomer here. We have Hara and Anissa Aulia tied in the first place. Also, Aulia Hasanah and Nuna Zatun tied in the second place. All right, let's go to the next question. The 6 questions. Which one is not including a sea cucumber? Remember, sea cucumber is not a plant, it's an animal. And the answer is Polycarpa aorta. Let's see what happens. Oh, the first place is still Hara, but Nabila is closing in at the 315 points. Nuna Zatun dropped a bit to the 5th place. Let's see. What is major secondary metabolite isolated from Indonesian soft coral? Okay, which one is it? Is it alpha, flavonoid, xanthan, or steroid? Well, at least it is not paranoid, right? It was the answer is paranoid. And the answer is this steroid derivative. Let's see. Hara still leading with 360 points. All right, the next question. Which motif doesn't present in this compound? Glucocycline, flavonoid, steroid, or hydrosol? Which is a good test for your eyes. And the answer is steroid. There are no steroids over there. Oh, we have a newcomer here. Iqbal Wahyu is leading with 371 points. While Hara stay in the second place with 360 points. Second to last question. Which organism does these compounds isolated from? Is it sponge, sea cucumbers, fish, or coral? Well, definitely from kangaroo. And the answer is... I actually don't know the answer to this one, to be honest. Oh, it was sea cucumber. I thought it would be sponge. Never mind. And in the leaderboard, there isn't much change to this. There is only change in points, not in position. Iqbal and Hara still in the first and second place. Let's get to the last question. Can anyone make it interesting? Oh, it's a very simple question. Where do we get this compound from? Sea cucumber, fish, sponge, or soft coral? One, and the answer is sponge, just like SpongeBob. Let's see who can actually climb. Oh, Hara actually reclaimed her first position. Hara is in the first place with 513 points. Congratulations to Hara. And all the other quiz will be the result of the quiz will be collected as part of your assignment. If you have not yet joined the quiz, we will provide a similar quiz to you at the latest by tonight. We'll send it through Telegram and Google Classroom. So please join if you have not yet. Okay. And I remind you for today, you also have your SFP peer assessment that you have to do at least once a day. For those who have do the peer assessment, thank you. You can do another one if you want to. But if you haven't, please go to the Gather Town and do some peer assessment. Okay. The next is, I think Bu Vidya is the one next.
Yeah, I think I have some information. Okay, everyone, it's me, Vidya, again. So it's nice to see you all again in this diverse weekend of our summer school. I think it's a day 5th, right? So I would like to give some information regarding the one that I taught you before in the morning session. So from your SFP that you have submitted to us to the committee, and then you can also see the SFP in the Gather Town. So we select the best 10 and we invite the 10 presenters to present your SFPO research on Wednesday in the afternoon session, to be exact. So the presentation will consist of 5 minutes presentation of your research or your SFP, and then followed by 5 minutes for presenting your SFP, and followed by 5 minutes of Q&A. So the total will be around 10 minutes. So we will inform all the 10 presenters through email. So we will email the 10 presenters. So please kindly wait. I think, yeah, within this day or in after this summer school, we will try to email the 10 presenters. And for the selected presenter, you are also required to reply the email to confirm whether you want to present or not. But don't worry, for other participants whose your name are not in the list, don't worry, because we will also provide you with a Google form. So the Google form, so please complete the Google form if you are willing to present your SFP or research. So how do I know that I'm not the presenter, I'm not the 10 presenter? So we will also inform or announce in the Google Classroom. So please kindly check the Google Classroom, maybe yeah, after the summer school or later in the night, maybe before 8 p.m. or GMT +7. So you can go to the Google Classroom, and then we will provide the link for you if you want to present your SFP or research. So for Wednesday, there will be 10 presenters who will present their research or SFP. So the 10 presenters, so we select the 10 presenters, and also there will be some of you who maybe want to present. Yeah, it's kind of like a voluntarily, if you want to voluntarily sharing your experience or sharing your research to others, you are very welcomed, and we will provide you with the G form. So you have to complete or fill out the G form, and then and the G form will be shared in the Google Classroom. Hope this information is not doesn't make you confused. If you have any questions regarding this, I'm very happy to answer. If you have any question, or is this clear enough? Okay, yeah, so for some of you, if you are not in the list, if you are not in the list of the 10 presenters, and if you don't have any research, don't worry, especially for the bachelor student, maybe at the first or second year who haven't any research. Don't worry, you can also present and share your maybe your review on a specific journal or specific research. You are very welcome to do so. So yeah, please be you can fill out the G form that we will provide you later in the Google Classroom.
Vidya?
Yes.
Maybe also I have additional information for all participants. So since Wednesday is coming closer, right? And it will be our last or final days. Yeah, very, very sad to know, but anyhow, everything must come to the end, yeah? And since we have variety of participants with the country origins, I would be in my opinion, it would be great if in the closing ceremony we have the opportunity for participants, representing their countries, anything, around 5 minutes something. You can prepare video or something. I guess within this period, you don't have the activity within a group, right? So maybe the participant from Pakistan, you can join together with other participants from the same countries. Yeah, for example, Kanita and Pusakit and other member. And then maybe from Philippines or from Vietnam and so on. Yeah, of course, the number of the participants is not equal, but don't worry. The good thing is we give you the opportunity to promote your country or to promote everything from your country. It would be great, in my opinion, for our closing activities on the Wednesday afternoon. So if you agree with my opinion, it's very happy for me. So for me, it's a kind of the good reminder. Last year also we had really great time when everyone shared about their own country. So it would be great memory for us to closing our summer school. So if you don't mind, please work together with other participants from the same countries, and prepare something. Everything will be, how to say, very welcome, to be presented here. Maybe 5 minutes enough, I guess, Vidya. And then you can sing, you can whatever, you can prepare video, whatever. Just for having fun, for all of us to close our summer school for this year.
Yes.
Hopefully you agree. All of you agree with this.
So it doesn't have to be a video. Maybe some of the participants want to sing live, or
yeah.
Okay.
That's also very welcome. That's also very welcome. Or whatever. It would be very welcome. Because
I would love to hear someone sing their traditional song.
Yeah, it would be nice. It would be very nice. Also from Indonesian participant, it's also very welcome. So I guess 5 minutes is enough for brief introductions or brief performance. Anything. It would be very, very happy. Because I observed since the first day, not all participant has the opportunity to open camera or something, and then when they prepare something, I guess within these 2 days, it would be more than enough to prepare some really short project really short project for the closing ceremony. That's all maybe from my side.
Okay. Bu Vidya, do you have anything else?
Tokir, give the comments here. Very good. Can we represent Pakistan in video about culture? And he's like, oh, of course. That's the thing that we would like to know, because we don't have any good opportunity yet to visit Pakistan, is it? Since we have very long restrictions to have traveling. So yes, very welcome, Tokir. That's very, very great idea. This one of the example, I guess. You can present a kind of video that also we give you every day. Wonderful Indonesia. You can display the video and then you can provide the brief descriptions about what is it. In the video. Okay. Also from Nigeria, from India, from Japan, some participants. Very welcome, I guess. Put short reminder on what to present on the last day through the class. Okay. Maybe PD and all committee members, so please kindly remind all participants in the Google Classroom. So no one will miss it. About the 10 best presenter that who have to present in our last session in the Wednesday afternoon. and as well as the great opportunity for the representative of each country origin to present something in the closing ceremony. I guess it must be fun for us. We have how to say a great engagement, to comment, to enjoy any kind of the presentation that you prepare regarding to your country. Yes, I guess, okay, Kanita, thank you for your great response. So hopefully other participants from Thailand, from Malaysia, from Vietnam, from Nigeria, from Philippines, and other country. I'm not sure if the participant from Australia also joined with us still, or from Germany. Maybe the committee need to check the attendance. But if still, it will be also great for us. So just in case, for representing each country, we have around 10, if I'm not mistaken, in the list, include Indonesia. So it would be enough time, to share each other about the interesting things, derived from your country. So I guess that's all. The announcement from my side. Fidi, do you have anything else besides the 10 best presenter?
No, that's it. So please kindly check on the Google Classroom. So we will announce and we will provide the G form if you want to voluntarily present your research or your SAP. And then we also announce on what to as Dr. Fitri said before, that we want you to make maybe short video or other things that you could do to present your own country, so you can work within your group from the same country to present a video maybe, or live performances or something. Something like that. We will announce the all information through the Google Classroom.
Thank you, Bidi, for summarizing.
So that's it, Pak Hafiz.
And the attendance links already here? Okay, this one. This is the most wanted, is in every last session. Attendance link. Everyone asks, where is the attendance link? So here you are.
All right. Thank you very much to all of the committee. This is the end of the session for today. We will let the Zoom meeting run for a little bit. I think about until around 4:15. I think. So you can fill your attendance list. Okay. Thank you for your attention, and see you tomorrow. Please do cover tomorrow, because tomorrow we will be talking about computational chemistry. For those of you who have not yet familiarized yourself with computational chemistry, I myself is a computational chemist. But the difference is I work in environmental computational chemistry, not natural product computational chemistry. But tomorrow we will talk about natural product computational chemistry, and another one is application of computational chemistry on trying to solve the COVID-19 problems. So please do come tomorrow. Thank you.
All right. Thank you, Pak Habib, for your great performance on sharing the sessions today, from the morning to the afternoon session. So everyone, the attendance link already list on the chat box. If you couldn't find it, just scroll it up. And also the reminder for all participants, yeah, after this meeting. So please kindly join to the Gather Towns, yeah, to fulfill your requirement to make a review, yeah, for today. And don't forget to submit your review result in the Google Classroom, yeah. Okay, so please kindly fulfill the attendance link, yeah, and have a great rest and also a great day. Don't forget to come back tomorrow. As Pak Hafiz mentioned, we will have the special talk tomorrow about the computational chemistry. Yeah, maybe several kind of the modeling system. Yeah, for the organic compound or natural product chemistry. Yeah, will be introduced by our great speakers. We have Pak Kaka. Yeah, very say famous and a legend of I guess legend person in Indonesian Chemistry Association. Yeah, we call it Pak Kaka, but his name is actually very long. Tomorrow Bumita will assist you, yeah, on the morning session as well as the afternoon session. And in the afternoon, we will have also the very talented and young great scientists from Pajajaran University. I've been met before with him. Deep impressions. Yeah, he graduated from Europe, if I'm not mistaken, and yeah, very wonderful. Yeah, his work on computational modeling, especially on medicine, if I'm not mistaken. Yeah, and some of how to say natural products. Yeah, really amazing. Yeah, so don't miss it. Make sure you can join again with us tomorrow. Yeah. If you have any curiosity, any lab questions about how computational chemistry contributes, yeah, on natural product chemistry research and organic chemistry in general, you can deliver that kind of question tomorrow to Pak Kaka as well as to Pak Muhammad Yusuf. Yeah, really glad to have you again tomorrow and to meet you tomorrow. Okay, thank you all. Thank you. Thank you. Yeah. Have a great day. Thank you. Thank you. See you again tomorrow. Thank you, Fanny. Thank you, Fanny. Make sure you come back tomorrow, yeah? Yeah. Thank you, Fanny. Hey, best speaker, Fanny, for colleges in Universitas Negeri Jakarta. Everyone stay there, yeah, and also in the great health. We will close the thing within 2 minutes. Yeah, so make sure for the one who hasn't yet filled the attendance link, so please kindly proceed it. So at 4 p.m. I will close the meeting. For all committee members, thank you so much. Yeah, it's awesome. around 16 participants here, so maybe around half, because almost 7 with me, the participants here. Oh, yeah, Mita already left. Oh, Mita still here. Okay. Yeah, tomorrow, Zara, Yusmian, 40 Cham, Pian, Tapi, yeah. And I forgot to mention, for those who already submitted some applications, so please kindly and patiently wait. Yeah, we couldn't proceed yet all of your applications due to our complete lockdown in our university. So we couldn't reach the office, as well as how to say meet some responsible person, yeah, regarding to the scholarship and so on. So yeah, just kindly wait. I will provide you the update after the lockdown period, yeah, is finished. So to Tikir and then to Saman, Kanita, yeah, and some participants who then me, yeah, through email some application as well as your CP. Yeah, it well received, yeah. And then I've been collecting all information. So yeah, just kindly wait. Yeah, kindly wait. I will proceed all of your applications after the lockdown period is finished. Yeah. Okay. Thank you, Zara. Thank you, Safi. Thank you, Putri. Nur Afika mana? And also Dang Kwok too. Yeah, Lofar as well. And also for Tang. And yeah, of course, my beloved committee, Vicky, Amelia, Janisa, and also Mita. Now it's time to close the Zoom. So thank you so much for all of your excellent effort today on organizing this meeting. And see you again tomorrow. Thank you. Assalamualaikum warahmatullahi wabarakatuh. I'm closing the Zoom meeting now. Okay. Okay. See you. Bye bye. See ya.